Good morning. Excited for today? I can see the excitement that you all came almost 15 minutes late. Anyways, so today is the last day of this GIAN session on precision fermentation for sustainable manufacturing of bioactives and industrial biochemicals. I hope, and I should not say I hope, I should say I sincerely hope that you would have learned something new, if not at least refresh the old part, whatever you have learned before. In today's session, we are going to discuss the case studies. The way we had decided that we were going to approach about the case study would be that I will provide you with the case studies. There are two case studies on the plant protein and the microbial protein. So, you will have your case study 1, case study 2. So, in the first session, we will work on the case study 1. You all had the opportunity to divide yourself in groups, work as group to come up with a solution on how you are going to produce the specific product. You might be using the same approach, might be using a different approach, but you will be getting an opportunity in today's session to talk about your approach. So, as we will go through the slides, when your team's slide comes up, you will have the mic to talk about your approach in producing the product. Simple? We'll start. So, the very first case study was developing a plant protein. The plant protein that I selected for you all was monelin. Now, monelin is a sweet protein, which is a single alpha helix packed against 5 antiparallel beta strands and beta grasp fold. Now, it is actually two different proteins which are connected to each other, like two amino acid sequences. It can be a bigger protein, it can be a small. There are monolins with only 50 amino acids, there are monolins with 84 amino acid groups. It's 1500 to 3000 times sweeter than sucrose, so the glycemic index of it is really low, and it's really good for diabetic people. Digestion has no impact on the blood sugar level, but the only concern with monolin is that the heat treatment over 50 degrees Celsius at low pH denatures monolin protein with the loss of sweetness. So, there has to be an approach on either changing the structure of monolin through genetic mutation, or you have to encapsulate your final product that you get to make sure that it can be used as a food additive. Encapsulation is just an approach which is commonly used in the food industry when we are working with heat sensitive food ingredients. There is a lot of research that goes on in the field of encapsulation. So, that is the reason why I have been talking about encapsulation in the past, because I am not looking at genetic modification of the protein. I am looking at using the native protein, how you can still make sure that it is used in the food industry as an additive. But if you have decided as part of your own process of modifying it, I am okay with it. So, it is obtained from serendipity berry, which is commonly grown in Africa. And one of the hosts that I had proposed would be a yeast, Pichia pastoris, which can be used to produce this on a large scale. When you are working with the yeast, the type of reactors which mostly is used would be a fed batch reactor. If you are looking at increasing the production of protein, there are also batch processes that I have seen. There's a couple of research papers that I have seen. There's a company called as Amai, I might be wrong, Amai or Amaya, which is in Israel. They are the one which is promoting this particular sweet protein, monolin. They are commercially producing it and they are selling it in the market. You will also be, as part of your case study, you would have been talking about the purification step, and you were also supposed to do something called as techno-economic analysis. Now, TEA is a big word and it's a big job to work on, but the way it works is that you have to define your process first, the production process. The idea is this gives you whether to invest in the required capital and what would be your return on investment from the product that is manufactured at the very end. So, it does take into account the cost of your raw material, it does take into account the cost of the process, but it also has to take into account the cost of unit operation. So, how much will it cost to have a fermenter, which is a 50-liter fermenter or 500-liter fermenter or 1,000-liter fermenter that you are going to use. You have to look into the market cost of procuring it, installing it, also the building in which you are going to put, what will be the cost of that. Everything has to be taken into account when you are defining the process of producing monolin. I know it's a big ask, but I just wanted you to at least have a chance to start working in the direction of techno-economic analysis, because if you really want to start your own startup as an entrepreneur, having an idea of how to estimate or how to calculate techno-economic analysis would be really, really good. Okay? So, we will start with the very first slide, switch shift.
Good morning all. Can I sit and or stand?
Yeah, you can sit.
You can give the first slide.
The first slide? Okay.
Yeah. This is Rajesh, Singh Rajesh from Chemical Engineering Department, research scholar. So, I am the one which who haven't no knowledge in the bio and all these things. So, because basically I am from mechanical and this chemical department and working with respect to these approaches. And coming to the name sweet shift, why I kept means, so actually we want to create one more sweet as an alternate to the sugar or the sweeteners, artificial sweeteners. So, that's why I kept a sweet shift, and it's good to keep double S as in chain like that, link like that. Okay, so and the case study and the one is the monolin. So, a low calorie sweetener. So, and we gave in a line such that sweetness redefined. Of course, even the what I did also that I made it the redefined from the SAR presentation. So, that's it. So, coming to the second slide. So, this is the main. So, where at first I searched for alternate for this fruit. So, whether India can have any alternate with respect to any residues like that. So, then I thought to use whether any like some mangoes or those fruits are there, whether they can have an alternate like that. I checked it, but I didn't get good appropriate. So, then I went back to the same the fruit which was tree which was there in African tree. So, the berry. So, then the first thing the thing is gene modification. So, basically the chain and all this I told I can't get understand that one. So, that's why I simply kept a gene and the amino acid, and I took the yeast as the same, and I gave the yeast. So, why? Because as I read in the basic knowledge here and there in the some papers or somewhere, they said yeast can give better in production if you increase in large scale for these aspects. So, then I selected yeast than the bacteria. So, then when we did the gene cloning with respect to this gene and this yeast, these are the operating temperatures and parameters. So, the functioning parameters that we can give, like the carbon sources, and the temperature can be in 28 to 30 degrees centigrade, where the pH we can maintain 5.5 to 6 and the dissolved oxygen should not cross more than 50. So, it can be 30 to I gave 30 to 50. So, and the carbon source like glucose and the nitrogen source like amino acid. So, I gave all these as an outline, because being I didn't went into the deep. So I gave it as an outline because some papers mentioned glucose in deeps. They gave some other as an advancement some there. So but I mentioned ammonium salts and some added minerals like some minerals they also mentioned. So but they didn't mention those. So just to give an outline. So the next I went for the selected for the batch reactor. So why batch reactor I mentioned there for the controlled nutrient feeding. So even as in the presentations we had about that, if you have any one inlet such that we can give, so it will be best one. So that's why I selected the fed batch reactor and for the good density growth. So then coming to the the one more section I made is the purification and getting the thing. So here the as I went on to research regarding what can be, whether it is intracellular or intercellular. So then some said that it will be in intracellular, so in between inside the cell. So that's why I selected intracellular, and then this is the new one I learned. So that means how to extract it. That means the purified. Being one of my lab mate is from biotechnology, he explained me so how we can get it. So he helped me in this. So that's why I mentioned those cell lysis. So breaking that one and the protein extraction and purification with the help of some chromatography, ion chromatography, and they mention, and then you can obtain the concentration with the whatever obtained. So that can be the saturated or crystallar like that. So then we can go for characterization, and this purified one which dried or something, so will be the monoline production. So this is the one. So like this an outline for making monoline with the fruit residue. So this is the making line. So coming to the next slide, which is then I went for the techno-economic analysis, and I went on a preliminary study regarding whether it can be feasible in making this product. So then I went with the factors. We consider the factors like raw materials, the cost of yeast, and the carbon sources, and the gene modification. Suppose if you give to any company or someone, so they will do the gene modification, how much, whether they will they will charge outsources. So the gene modification we can use RNA laboratories. So then, and the energy cost for the aeration or for the stirring for one batch. Actually these are made with respect to the batch. The energy cost and the purification cost, the filtering and centrifuge only I mentioned here. If you you can add chromatography and like this also we can, but I mentioned these two only. So because at that point I don't know that this ion chromatography will be there. So after that my friend said. So then the capital costs such as the investments for us reactors and the accessories like the sensing systems and the space for the setup and the infrastructure where I kept like this. And the labor costs where we added the skilled labor and semi-skilled labor. These two in general what much we can give. So based on this, then the cost breakdown, summarized it like this. For the production for the one batch, the substrates which can be like this, and the energy used will be 8,000 and the labor cost will be like this. So whereas, suppose if you write this for an student project proposal, so then you can write for 10 more than 10 to 12 lakhs, so that we can make one project. So then I went on thinking, went for the cost, how much cost the monoline for 1 kg will cost. So then it costs, so when I said in respect to different things, so it costs about, they are saying about 30 to 50,000 per kg they are mentioning. So this is the huge, I shocked with the price. So then I compared, so the next slide. So then I compared with the others sweeteners, alternates. So whereas any alternates are there. So then monoline as this much price. So then a stevia, so which can give, they are coming in 5 to 15,000. So and moreover they are saying if it is a stevia is in liquid form, then also it will very less cost, nearly 5,000 in that. Okay. So then then again, so is there any alternate to stevia? So then I went for the this sucralose. So then These are the charges. So then finally, as in these analysis, I am saying so. So whether it can be cost effective or the thing, I mean analysis, what it means. So it's an low calorie sweetener. Okay, very good. And it is a premium one. So and those who are interested in with respect to the pharmacy or health conscious food, then they can approach it. And how then the next thing is how we can fight with the others. I mean how we can give the competitive pricing strategy. So then we have two pricing strategy, like if you give it for the right application in the beverages or health conscious customers and the consumers, then okay, they can when then only we can have it a better alternate with that one. So as per the as per the my knowledge, and moreover, so this was everything was done on the basis of the study, primary study. So the one who has zero, I mean limit knowledge in the these aspect as an industrial point of view or someone, so who did it. So because so if you go for investment or someone, they don't know nothing, they will ready to put the money and they will look for the investment, I mean returns. So then as in that case of study, I made this like this. In the technical point of view, okay, of course it's great and well and good. The country like India can do it, means no, we can't afford it. So if you have any more upgradations in gene and the production is increasing, if you go in the large scale, and then only it can be good in the country like India. And thank you for this.
Thank you so much.
Thank you,
I
sir.
really appreciate the presentation that you have done. It's a really good effort, to be honest. I'll give you an A star. I have really hard time to design a star, but
And of course,
I'll really give you a
and
A
I
star.
failed in making a team. Sorry. So actually, this is the team thing. So if I made it a team with respect, then maybe it can be a triple star or double star.
You want me to give you a double star? I'll give you a double star. I'll give you a
Not
triple
at
star.
not at all. The rule is rule. So I missed that rule. So it's the solo thing. So sorry.
No, see, my intent was at least for you to make an effort to understand what the process of utilizing precision fermentation, the concept of precision fermentation is. When you're talking about gene splicing, when you're talking about taking the genetic material from the plant and putting into a yeast. Before we get into that, I will go back to the very first slide. Do you all have any question for Mr. Rajesh? When you have the question, just raise your hand. We will pass on the mic to you.
Like sir had asked us to make it thermally stable. So like at which step you are actually making it thermally stable? Because this is a very generic outline for monolin production, but we have to make it actually thermally stable, and we have to check whether the protein is thermally stable or not.
For that, I added on. Sorry, I didn't added that one. So for that, as Sir said, suppose you use it in tea. In tea. Suppose you want to make it in tea. You want to add this sugar. So then, so as less than 50 degrees, you can make the tea hot. Okay. Then if you add in less than the temperature, then it is good. Okay. So even this is the one thing I can suggest. Suppose like hot water, which should not more than 50 degrees centigrade.
No, I understand what you're saying. Okay, when you're looking at the process from the food industry perspective, every product that you buy, specifically beverages, they have to be pasteurized. Now, when you're thinking about products like monolin, which are pharmaceutical grade purity, they don't require to be pasteurized. So, after the pasteurization process, they can be added to your product. So, the temperature will not matter at that time. And most of the juice, if you are looking at fruit juices, they are kept at lower temperatures, they are not kept at very high temperature. Even if you are looking at the Tetra Pak, they don't ask you to put it outside in the sun, like not to go beyond 30 degrees Celsius, right. So, there are possibilities of using it without encapsulation. My intent of bringing encapsulation was to see if you have any idea of approaching it that way. The idea of tea is something that we do. Not everybody makes the tea the way we do, right? So, it's a very different way. Any other question for Mr. Rajesh? You are free to ask. It's an interactive session. This is the only interactive session.
You said that you searched about Indian sources, but you couldn't find that. But there are Indian sources which can produce monolin as well, like anjeer, kakrol. So why didn't you use something like that?
Actually, in my point of view, I search for the feedstock waste. Okay, so usually in generally the anjeer having waste will be very less. I am thinking. So actually not.
So you were using, so the raw material that you wanted for your
For this
protein was supposed to be a feedstock, like a
feedstock
waste.
Suppose if you
Okay.
eat the mango peel is the waste. Okay, so definitely everyone will consume the mango everything, and you will leave the peel. So like that, peels like mango peel or some like that, I searched with respect to that waste.
Okay.
So maybe they may be there.
Let me put another alternative process or alternative method. There are research papers, if you would have looked onto monolin, where they have taken the gene that actually produces monolin, they have tried to put that into tomato plant. They are trying to see if the tomato can have the monolin protein in it, the sweetener in it. They were able to get, let's say, 15 or 20% in that, but it's not a very successful process yet, because it's still a growing field of research. It's a very new idea. The concept of using sweet protein is still very, very new, but it's a good approach to look at. And thank you for bringing the example of anjeer. Now, anjeer is considered to be really, really good for your health, right? From the Indian food perspective. I am a fan of anjeer. I do eat that a lot, specifically dry one. I don't like it to be soaked. Many people prefer it to be soaked overnight, then eat the next day. Thank you. I prefer it dry. So there are possibilities of where we can source our monolin. The concentration will matter the most. So there are a couple of approaches. Either you take, so if you want to extract monolin, you can extract it from the plant source directly. We did had one example yesterday where we were extracting everything from the plant source directly. We can do those type of things. So if you want that, you have to grow it in the plant in larger quantity, or that particular plant should have a shorter growth time. So you are able to produce it on a repetitive basis, month after month, or let's say weeks after weeks, so that you can have enough production, but still requires a lot of land. When I say land, that means fertilizer. So again, you are looking at the GHG. So if you will start doing the LCA, life cycle analysis, for your process, that would not be very supportive of the idea until unless you are not using any chemicals or pesticides or things like that, right? Now, I do understand the reasoning why you chose yeast. Why Saccharomyces cerevisiae, if I may ask? Because that's the most common yeast that you are aware of.
Even I know only two. One is E. coli, and
E. coli
this one
and
E.
this,
coli,
okay.
and then this one. So I took these two only.
Oh, no, it's a good idea. You can definitely go for Saccharomyces cerevisiae because, excuse me, what will happen is because people are already aware of Saccharomyces cerevisiae that has been used for baking purposes. So if you're producing a product from Saccharomyces cerevisiae, people will not be offended or will not be against the idea of the product. That's a good thing. What would be, like, for your process of the fed batch, what do you think would be the limiting factors? Because you talked about dissolved oxygen to be 30 to 50%. So you're talking about the OTR, or you're talking about the OUR, like oxygen transport rate, or you're talking about the oxygen uptake rate?
Taking rate.
The uptake?
Yeah. So to use it.
You only have like 30 to 50%.
So just I mentioned the number. So as per the, so it should not use that much. It should not give resolve. not at all.
So you don't want the oxygen to be there for the yeast to grow, or you want the oxygen to be there for the yeast to grow?
Oxygen to be needed.
Oxygen is needed.
Yeah.
So you have, do you want to provide ample amount of oxygen for the yeast to grow,
So
or
it
you
should
don't?
be with respect to that much only.
Whatever is
not
required.
ample.
Okay.
How much?
And for the concentration process, I do understand that you have mentioned that you might not be aware of many things, but
Yeah.
what do you think would be the ideal technology that you can use for concentration? What is the most common technology that is used for concentration?
Not an idea. Anyone can.
How do you get salt, sea salt?
That is drying, crystalline. That's
Drying is the
the process.
easiest and the most important unit operation that is used for concentration. Now, when you are working with protein, you cannot do high temperature drying. We will be doing a low temperature drying, specifically freeze drying. Now, freeze drying is a very costly process, but it is used in the pharmaceutical grade products, right? Because anything and everything that you start working with recombinant DNA and the products that you make out of these, they are generally purified up to that pharmaceutical grade. The other thing that can be used to reduce the overall cost for your process would have been use of plant residues as the source of carbon for the yeast to grow. So that can also be used because if the cell growth was the only factor, you can, I think solid state fermentation would have also worked for you. If you have your crop residues and then you have your culture over it, you are just providing the nutrients as drips on it, you might be able to still produce and still utilize the carbon source from the feedstock. It might be a slow process, but it's a process that can be used. Just a suggestion. The other aspect that I found really interesting would be the cost that you came up with at the very end. 30,000 to 50,000 rupees per kg, that's pretty huge. And I'm not surprised monolin is going to be that costly if you are producing using recombinant DNA technology. You can only reduce the cost if you are going to reduce the production cost by using feedstocks, plus if you are able to utilize, if it would have been an extracellular product, it would have been cheaper to purify because you went into cell lysis, separation, all of that will add cost to your process, but it's a good effort. Thank you so much.
Thank you very much for the opportunity.
Now, the next group would be the procatalysts, procatalysts. Your turn, sir. Who's the spokesperson from your group?
There is actually two spokesperson. Means
That is fine.
That's
If
fine.
the whole group wants to speak, they can speak.
Okay.
But don't speak at the same time.
Okay, sir.
Okay, so you want me to change the slide?
Yes, sir.
Why procatalysts, if I may ask, before we go forward?
So there is no like such reason to like, it's just come random.
The team name is Procatalysts because we are basically using protein here.
Okay.
So protein as a catalyst. So that's why it's Procatalysts.
Okay, now because I got really confused because if you are like pro inhibition, pro.
No, sir.
Like
it's
that, like-
protein. For pro, it's
Okay.
protein.
Okay, got idea. Nothing political here, right? Okay.
So, good morning, everyone. So this is just the introduction of what is actually a monolin with sweet protein, around 1,500 2,000 times sweeter than sucrose, as well as those naturally available that's derived from natural sources like stevia and all. It's quite sweeter than most of them. So molecular structure consists of mainly of two polypeptide chains, A and B, connected by non-covalent bonds. Consists of beta crash fold, as Sir has mentioned in his slides. There are two types of monolin that is considered. For food grade, is double chain monolin, which is basically a heat-sensitive natural form. It does not exist in a functionable form at a temperature more than 50 degrees Celsius, and a single chain monolin, which is known for its enhanced heat stability, created through genetic engineering process. It is a protein model for research on test receptor interactions, aggregation, and folding. This is basically the introduction of the monoline protein. Let's move on to the upstream processing of the protein. So this is, you can see in the picture where I mentioned the strain that we have used, the BL21D3, and below you can see the PET duet factor. So here you can see this is the most cost-effective procedure and scalable procedure we have found, which from the host selection procedure, E. coli BL21D3 is chosen because of its capacity to produce the heterologous use concentration of heterologous production expression. Then most of the others is like other host system like Pichia pastoris or like yeast or Saccharomyces cerevisiae. Then is codon optimization, then insertion of the PET duet vector with two restriction enzyme sites. I have mentioned here NE1 and BZ1-2. So we used to clone the genes of the saints A and B separately. Then after that, transformation is conducted using the BL21D3 cells. After that, sir, move on to the next. Can you, sir? Okay. So after transformation, for insertion of the foreign DNA, its heat shock is induced, selection through ampicillin containing LB media or transform cells. After that, similarly the procedure continued, seed culture inoculation. The precultures were found. They were incubated at 37 degrees Celsius. Scale up was induced, which consists of 10% of the seed culture or the fermentation volume. And then medium optimization as well as is conducted for proper functioning of the next step, which is the fermentation. So this follows the fermentation slide, which I begin. So fermentation is basically done. Why it's done? To observe the efficient and scalable and economically viable synthesis of the protein on a larger scale. So there are like what factors that matters. Like I've considered medium the nutrient sources that matters the most. In carbon, glucose is considered because E. coli metabolizes quite well, as it's well known. In nitrogen, yeast extract, because as for many research paper studies, it's abundant vitamins, growth hormones, and amino acids, and best suited for E. coli. Peptin for strong development for similar source of peptides and nitrogen, and some traces amounts of salts required for efficient enzyme activities like calcium chloride and magnesium sulfate. So as Sir has mentioned in his slides, for this particular technique we are using E. coli, and so the state tank reactor will use the process of submerged fermentation for efficient nutrient delivery and fed batch fermentation, because in fed batch the substrates that are used by the E. coli or cells will not be used rapidly, so which will prevent rapid utilization of glucose and exhaustion of our substrates. It will gradually improve, which will enhance the protein or product production at a larger scale, and which eventually producing the required concentration of protein. So the parameters considered for fermentation is 37 degrees Celsius, and the pH at 7. It's kept near neutral level, some 6.5 to 7, but I mentioned 7 since it's most favorable. Aerobic development, the DO, oxygen saturation concentration, is kept at around 30%, and aeration and agitation, according to our model, is kept around 300 to 500 RPM, and 1 VVM, or volume of air with aeration rate. So after that, after fermentation process, after rapid growth we have observed, the induction was conducted for the protein expression, intercellular protein expression. To that, IPTZ is used, which is basically a allolactose for protein induction. This is uses substrate. So again, that for this one induction process, the DO, the diesel oxygen, is maintained at 20%, with less concentration done. And then IPTZ which is used is at a concentration of 250 micromolar. After that, if there is any foam production because of the protein formation, and defoaming agents were used to control the foaming. And this is followed by the cell harvesting, which is usually done through centrifugation at a rapid rate of Z, centrifugal force, for 10 minutes at a temperature of 4 degrees Celsius, which keep the protein at most suitable to prevent degradation of the protein, and then storage at minus 20 degrees Celsius for later utilization. So after this process, you can see in the picture. Sir, can you move to the previous slide? Oh. This you can see in the picture of this one, submersed fermentation, how the whole process is connected, how media preparation is connected to media sterilization, and then how it's the whole controlling of parameters are conducted for efficient fermentation procedure. So this is followed by the next steps, which will be discussed by my colleague.
Thank you, Ashutosh. The next part is extraction and purification of monolin. So this we can say this is basic outflow of how downstream processes carry on. But in this process, we will basically follow on the industrial scale and which kind of equipment, which kind of process, columns and resins we can use at industrial level. So first, once we get the cell from cell harvested, for cell harvestation, as Ashutosh said, we can use a centrifuge, like Alpha Laval is there. They have their product like BTPX, which have a flow rate 2000 liter per hour, and which requires a 15 to 20 kilowatt for energy. So next, after centrifugation, we as our product is intracellular product, we need to break it to get our protein, to extract our protein. So for this process, we can use a homogenizer. Basically, homogenizer equipment that pressurize our feed, and it's an impact on the metal plate. So cell can rupture and our protein can get into the media. So that's why we use a homogenizer. Like YO Cell Bio is one company that which can produce the homogenizer. For like they have a feed rate 120 to 150 liter per hour, and they work at 1800 bar pressure, and the power is required for 11 kilowatt for this equipment. We need the incubation for the settle down the impurities or precipitation, and at this step monolin does not precipitate, only impurities can get precipitated on the basis of gravity. So again we need to centrifuge to remove the impurities, and our protein will be stay in a supernatant. So this supernatant we can use for the further separation, which is a chromatography. Basically we are going to use ion exchange chromatography. So basically in ion exchange chromatography you are going to use SP Cephalose resin, which is a weak cation exchange chromatography, and which is we can get a 90% of pure form from this chromatography. For this purpose we can use Acta Avant for lab scale, Acta Pilot for a pilot level scale, and Acta Process for the industrial scale. And column we can use a BPG column at a 25 centimeter height and 500 millimeter for a pilot scale. And for elution purpose we can separate out our protein on the basis of salt concentration, and we can elute out the protein at using a elution buffer which contains the 100 millimolar NaCl. And the elution is on the basis of the UV absorbance. The graph shows UV absorbance at 260 to 280 nanometer. So after that we will capture our protein of interest, but with that we can also get another protein also. So we need to further separation using a gel filtration chromatography. For gel filtration chromatography, again we are using same instrument like Acta Pilot or Acta Process, but here we are using size exclusion chromatography resins. Like for example, we can use Superdex G75 for lab scale, and for pilot scale or for industrial scale we can use Cepharex G25, which we can separate the molecule on the basis of molecular weight, which can we separate the molecular which more than 3000 molecular weight. So for these purposes we have used Exichrome 200 millimolar column. Again we can use a BPG column for here also. And then we need to ultrafiltration for the making the product, to make the product concentrate, to remove the excess water content or excess buffer content. We can use ultrafiltration. Here we can use the ultrafiltration different size plates. Following by the ultrafiltration we need to lyophilization. Once we get our concentration product, which is more about to 98 to 99% pure, we need to lyophilization to make it API, active pharmaceutical ingredients. For API we gonna use lyophilization system India's model lyophilization. We have a capacity to lyophilize at a time 200 liter, and after the lyophilization we can get the active API, which we can be further store in store, and we can further use for the next purposes. Next slide. So next, this is an overview of the downstream processes. The simple protocol for purification of double chain monolin it involves two chromatographic steps, so we can get highest purity that we can achieve. It does not use any protein tag or expression for a purification. The protein, purified protein is more than 98%, and we can analysis it using by the SDS page. And the yield of protein is up to 40 mg per liter of E. coli culture is obtained. Next is techno analysis. We'll explain. Parthivi.
In the first step, we have used a host organism which we have genetically engineered, and the organism used here is E. coli BL21 DE3. DE3 is the vector that is being used. So approx cost that has been taken for the plasmid synthesis, gene editing tools, and the strain development is around $5,000 to $5,000 to $10,000. And for media formulation, for all the carbon source, nitrogen source, the trace element salts and all, approximately for a 100-liter batch, we have taken $1,500 to $2,000. And for inoculum preparation, for the seed culture, for the to cultivate the seed cultures in the shake flask or the seed fermenter, we have taken approx cost of 500 to 1,000 for a seed fermenter preparation. And for fermenter selection, we have taken here stir tank bioreactor, which is of 5,000 to 10,000 liter scale. That is a industrial. That is for the industrial production, and it is a one-time capital investing. So we have taken approx of 2 lakhs to 4 lakhs dollars. And for the fermentation process, the parameters like temperature, pH, maintenance, and the dissolved oxygen, etc. For a batch, we have taken around $5,000 to $10,000 per batch. And for cell harvesting, centrifugation process, for the centrifugation process, we have taken approx of $500 to $1,000 per batch. And for cell lysis and protein extraction, $1,000 to $2,000 per batch. And for purification, like chromatography, like ion exchange and the gel filtration, it is around 5,000 to 8,000 per batch. And for polishing and formulation, for like ultra filtration and all, we have taken around, it will be around 1,500 to 2,500 per batch. And for the drying and packaging, like for lyophilization, as it is very costly, so it's around 2,000 to 3,000 per batch. And the final cost, that is the purified monolin, that is to be sent to the market, the estimated price is around $15 to $25 per gram. This is the purest form. Thank you.
Cool. Thank you. Now let's get back to the very first page. Any questions? If you can pass on the mic, please.
My question will be like, as far as I know, that bacteria, if we use bacteria as a host organism, then it might lead to protein misfolding, right? Because it doesn't have proper post-translational modifications. So what steps will you take if you are using a bacteria as your host?
Actually, there is one step. Once we get the bacteria after homogenization, there is one step called refolding, where we use basically we in industry use urea, EDTA, trees, such agents to refold the protein structure, where we give an incubation time with at a specific pH, at a specific temperature, for like let's say 15 hours, 26 hours for a protein to get a refold at what we want, and then after refolding we'll proceed for the purification. So there is one step refolding that we have not mentioned here because we didn't get a data for such this protein, but for other protein like insulin, glargine, there is a step like refolding where we can refold the protein as per our requirement.
Good, good. Any other question? At the very back.
I have one question. So E. coli is a gram-negative bacteria. So it has a, what is that, lipopolysaccharide enzyme it is there. So whether it will excrete endotoxin effect, so how will reduce that? Because of that only, we didn't choose that.
So if I can explain it in a layman way, E. coli can produce toxins. What will happen to your protein? Or those toxins will also be there in your product? How will you take into account of removal of the toxins?
So as it is a recombinant E. coli, so before using in industrial scale, we at least everyone should see how toxicity it can show, which any kind of toxicity can introduce in product. So I don't think it can show toxicity and it can have impact on a product.
I'm actually very, very proud for you asking this question on toxicity. When you're working with a very highly pure compound that has pharmaceutical properties or even can be used for nutritional purposes, you have to perform toxicity tests on animal models. Okay? You have to perform the toxicity tests on cell lines and evaluate if the product that is coming out after the process is done is good enough to receive the regulatory approval. Because any product that you make, even if you have all the TCAs and everything with $25 for a gram, it's a very interesting bargain. Even if I'm putting in, let's say, $40 million of investment to set up the capital cost, I will be making a hell lot of money with that cost, the value that you have for the product, if there is a market for it. The point would be, if it is toxic, you have to perform all of those tests to prove that it is not. Nobody till now had asked you that question, but that should be part of your techno-economic analysis, where you have to conduct all the lab trials before the product is even launched. The R&D and the food safety and quality, the QA aspect has to be included when you are looking at the capital cost, when you are looking at the production cost. So those things have to be included to a certain extent, but really, really good effort. One question that I have, another question? Yes, there is another question, please.
Sir, and it secretes low amount of protein. So in downstream process, the cost effect may increase that. So
what he is asking, like your production.
No, sir, because we are using E. coli, no?
Yes.
So it secretes a low amount of protein.
Yes.
I think so. So that in downstream process, the cost may increases.
Okay, how will you increase the amount of protein that E. coli will produce? It's a very good question. So this is the DNA that is actually producing the protein, or the E. coli is the DNA that is producing the protein. So you are transposing it onto the E. coli DNA, or you have the plasmid which is going to run by itself. What is the idea? The group needs to answer this. So are you putting the DNA in the E. coli DNA, or it is just a separate DNA that is going to be
plasmid is an extracellular DNA.
Yes.
In which we have using the strong promoters.
Yes.
Which will overexpress the protein the DNA of that protein.
Yes. So because you had mentioned something about the inducers, right? The IPTG. That inducer is for the plasmid to initiate the production. Can you, when you're doing the heat shock, can you just give one plasmid, or you can give multiple plasmids? Like, is it like one chapati per E. coli, like one chapati per person is like one plasmid per E. coli, or you can give multiple plasmids? It's a good question. There can be multiple plasmids.
But sir, there may be like due to like plasmid incompatibility might occur if you put, because of if there are similar power regions which helps them in replication. plasmid
Okay.
replication, it can induce incompatibility. As we have observed, like in E. coli, there are around 30 incompatible groups.
Okay.
And also if Staphylococcus aureus is used, so it's around 13 incompatible groups that have been identified over the last 15 to 20 years.
Okay.
So before it was not understood why it's happening. So after the analysis of to study the plasmid replication, so when they possess the same PAR region or lack the variation in the PAR regions, it often lead to like plasmid incompatibility. One will get expressed, or sometimes means it will lead like the protein which we want or will get suppressed eventually.
Very good. Like, you're right. I'm not saying that you can fill the E. coli with all the plasmids. You can have more than one plasmid, but it might not work. What would be the other idea? Can you give more of your sequence? Like if let's say your system is basically, let's say if the sequence of your interest that was producing monoline, if that is incorporated in the E. coli DNA, if you remember when we were discussing about that, so I will make a random cut in the DNA, I'll put my DNA of interest and then close, right? What if I can insert my interest of DNA at different locations? Because that would be a random, like when the promoter starts, it won't be that each and every sequence that I have inserted will get activated. But can I do that? Can I add more of my gene of interest in the E. coli DNA by slicing it at multiple locations, then connecting it? If I do that, I would be translating my protein multiple times, right? That can increase your production. But there are techniques that you can be used. So to answer his question that whether E. coli can produce more protein, there are these techniques that are used. But the problem is, historically, what I have seen personally would be yeast has always been the best choice for protein production. E. coli have a much better production rate.
Yes, sir.
So if you are doing batch, you would be able to produce more. So you might be able to have enough production to match your requirement. Any other question for the group? I'm not going to torture them more, that's why I'm asking. Yes, if you can ask in the mic, that would be great.
But sir, if we randomly slice the DNA, then what if we actually slice because we are randomly doing it, what if we slice an important
It's always
sequence?
random. Yeah, that is always there. That's why you have to do the selection after you have put the plasmids in. Not all of your bacteria will grow. Only few of them will grow. That's what happens.
But so then doesn't it make plasmid the better option?
It does. That's why we use plasmids, because they are the much better and safer way of producing or adding our gene of interest. So there are two ways. Either you put the plasmid and let it grow, like let it replicate and translate and produce your protein of interest, or you randomly cut and then put your gene of interest. But that's a slow process. It might get activated, it might not get activated. We were discussing this when we were talking about synthetic biology. Like if I put it anywhere in the sequence of the DNA, maybe DNA is not interested today to translate this zone of the DNA, and my protein was in that particular zone, so it won't get translated. So that happens. Protein folding, that was a very good question. One question that I had, heat shock is to generate pores in the cells so that the plasmids can enter, if I understand correctly, for electroporation?
Yes, sir.
Okay. So the way we add plasmids into cell is by electroporation, either you can, or by heat shock.
Yes, sir.
So you make the membrane open a little bit so that the plasmid can get inserted. Okay.
Yes, sir.
Good. This was good, and I would have really appreciated you would have mentioned the company's name on the side, but that is fine. With respect to the amount that you have mentioned, it's all in US dollars, right? Okay, so if you are representing, it's always right, 5,000 USD, because there is a Singaporean dollar, Australian dollar, Canadian dollar, so we don't know which one you're talking about. Okay. Any other question for the group? But great effort, really appreciate it. Thank you. Okay,
Thank you, sir.
the bluff team. Your turn. That will be down.
So our team name is Bluff, and it's basically Biotech Lab united with foodies.
Okay, so it's a pretty big group. I
Yes,
like
sir.
the team spirit.
So these are our team members, and we can start with the second slide.
Okay.
Yeah. So we have basically
So while talking about monilin, so there are a lot of artificial sweeteners are there in food industry. So the monilin is one among them, even though they does not have GRAS status, but even though there are studies are going on to improve their GRAS status, like to get the GRAS status in such a way that like it's a protein, it's a peptide, like so definitely there are a lot of studies required, like it do not have any side effect and all. So there are different types of species which produces this monilin, and we have chosen one among them which having the highest production capacity, the gene corresponding to the highest production capacity. And usually monilin have a temperature, like lower temperature stability, because it's a peptide. And as we know, in during food processing there are a lot of stages are ongoing, like as you know, pasteurization, sterilization, blending, mixing, agitation. So during this process there are chance of unfolding of the protein, as well as chance of denaturation of the peptide molecules during the heat processing. So there is a need of stabilizing the protein molecules. So the first thing is like we have to stabilize the peptides with respect to the higher temperature, and the second one we have to stabilize the peptide with respect to the processing. So either we have to encapsulate it, as well as like we have to encapsulate it, as well as we have to stabilize the peptides and should be withstand high temperature. So moving to the
Okay.
next.
I'm so sorry for the way it appears on the screen.
So and we have selected the specific species for the isolation of the gene, and this is the genetic sequence for the same. And we have used the CTAB method for isolation of the gene, and the after identification of the gene we have went for amplification, and the primer design is also needed. So the forward primer and reverse primer as given in this slide, and after that we will be amplifying and cloning into the particular organism, and cloning of the monilin gene to the particular organism. And in between, as we know, like there are different technologies that has been introduced, like even point mutation and other mutation techniques is introduced in order to improve the heat stability while modifying the particular sequence or particular amino acid, which does not cause any changes in the properties, whereas it will improve the heat stability. So we have constructed a CRISPR-Cas9 vector. We have introduced the particular gene into the CRISPR-Cas9 vector and transforming into Aspergillus species. As we know, Aspergillus species, which is highly secreting and it's extracellular, like it will produce the protein extracellular. So there is no need of further rupturing or destruction of the cell for isolation of the particular protein molecule. That's why we have selected Aspergillus. As we know, Aspergillus is commonly used in the production of acetic acid, and it's very, even there are a lot of designs are the bioreactor designs are there for production of acetic acid. So anyway, it is commercially available, and people is ready to accept whatever protein or whatever products which is produced from Aspergillus. So, okay. So design the synthesis of this guide RNA. After that, we construct the CRISPR-Cas9 vector, like clone the guide RNA into the CRISPR vector and insert the monilin gene. After that, then transform into the Aspergillus. So after that, we have to know like whether this Aspergillus, which is like it is the gene which is expressing or not. So we have to go for the culturing. So we will be verifying the gene integration, whether it's done in like the integration is done or not. So for that we will be we have verified. So we are going to verify this like the particular gene sequence is there or not by using PCR or qPCR or Western blotting. After that we have to go for the genetic modification for the heat stability. So for that we have used a specific mutagens, that is E23A, so which will transform that glutamine in the 23rd position to alanine. So there are a lot of literature which shows that, we forgotten to given the reference, even the reference which show that even once we are altering the particular amino acid like glutamate with alanine, so the glutamic acid is highly prone to lot of reactions once it is present in the monilin. So we can stabilize it, stabilize the particular peptide while altering the amino acid by alanine. So this studies shows that almost the temperature can be like the stability is retained until the temperature of almost 85 degrees Celsius. So it will be stabilized. It can be stabilized at 85 degrees Celsius as well. So most of the fruit and vegetable pasteurizations are in the range of like 80, like 70 to 80, that range. So definitely we can, even high acid food, there is no need of high temperature. So we can pasteurize it. So even we are introducing the monilin, that artificial sweeteners into any fruit juices, we can stabilize it by this method. So while producing by this particular methodology, after that we can go for the expression of the gene. So whether this gene is properly expressing or not in the particular host, we have to find out. So for that, after that, once we will be going for a small scale, then pilot scale, and we can go for the larger scale. So meanwhile we have to check for whether we will be getting, we have to standardize the procedure, or we have to standardize the growth condition for the aspergillus. And as per different literature, we have finalized that fed batch fermentation, like submerged fermentation, still tank in a still tank bioreactor is suitable, because we need a proper aeration. And for growth phase, the temperature should be 37 and pH should be in the range of 5.5 to 6, and the agitation the range of 200 to 300 RPM, as well as the aeration is almost 1 to 1.5 ppm. And during the induction phase, that for the production of the or the expression of the gene, so we have to change the temperature as well as the pH and agitation as mentioned here. And as we know, during the aeration we require proper, like during the aeration there are chances of forming, so anti-forming agent like either EDTA as well as mineral oil, both having food grade, so we can use this. As well as we require the carbon source and nitrogen source such as molasses and glycerol. So glycerol, which is required during the induction phase, whereas for the initial phase molasses is required. So initially molasses, which can usually containing the glucose, sucrose, as well as followed by during induction phase it will be utilizing the glycerol. And nitrogen source such as ammonia and ammonium sulfate and urea. Apart from that, we require different minerals for the we didn't mention exactly, but it's required for the enzyme activity. So during the downstream processing, as we know anyway, immediately, even it's a extracellular production of the particular product, so we can reutilize the organism, and that is one of the advantage here. And another thing is like next after that, for example, we will be going for ultrafiltration, and the membrane should be able to pass almost 10 to 15 kilodalton, more than in that range. So we can specifically extract the particular peptides. As apart from that, we have to go for ion exchange chromatography for the purification. So meanwhile, we initially after this expression of the gene, we have to go for HPLC for whatever protein it is producing, and we have to verify like if any small amount of other protein fraction, if it is there, it should not affect the quality of the monolin. So that also we have to check, and once it is, we have to verify. If it is not there, then there is no need of further chromatographic techniques to separate it, even though we can specifically separate the particular protein fraction. And then we have even, there are different methods are there. Either we can go for lyophilization, even though it is costly, but from industrial, different industry, they are, for example, in case of stevia and other low calorie or non-nutritive sweetness, which is commonly blended with maltodextrin. As we know, the glycemic index of maltodextrin is higher, but even though we are using in a very small quantity, so it does not even create any problems in the sweetness, because like it's almost 3000 times sweeter than sugar, so there is no issue. But even though we can use it in like thermal stable products, but we cannot going for sterilized food products. As we know, like even high temperature and sweet products are very less, like the combination, even whenever we are considering any food products like sweet and even except some dairy products, because this sugar itself giving the structure to the product, so there is no need of altering, or even we can't substitute this monolin with this sugar, because in that product the sugar itself giving the structure. So in case of, for example, gulab jamun and other products, we can't use it because it will affect the entire quality of the products.
Okay.
Even sugar itself giving the texture and other properties, as well as kova or whatever crystallized food products that give sugar will give in the structure. So we can't, even there is no need of using monolin in that. So that's the case. So that's why we can use it for different fruit juices as well as tea and coffee. So anyway, tea we have to consume, like we will prefer the temperature nearly like above 70, and like hot tea we won't prefer to consume at boiling tea. So we can use once the temperature which reach almost 80 degrees Celsius, we can add it, like we can give. But practically I don't know whether it's possible or not, but even we can use, and even it cannot be completely denatured, it will be stabilized, some amount will be stabilized. And regulatory, in case of regulatory approval, as already in previous slides everyone mentioned that there are a lot of animal studies, inline studies are required. It should not create any kind of other health effect to human beings or any mammals and other animals as well. So for regulatory aspects, there are a long and years of studies is required. So for that as well, we need a lot of capital investment, and we can go for packaging material like LDP or HDP. That is does not matter. For industrial scale, we can blend with maltodextrin as the best choice, as per my understanding, our understanding. So
okay.
So the cost evaluation, for substrate, it is almost even as we are using molasses, so the cost can be reduced up to a level. The next one is like for there are a lot of initial investments are there. So once we are transforming the gene into the particular Aspergillus species, there is no need of further capital investment once we have set with all. And so further on, like even capital for example, nowadays most of the bioreactors are completely automated, so manpower requirement is comparatively less, and the dollars which is represented in US dollar, like for the better understanding. And fermentation, that is we are expecting approximately 50 to 200 dollars per US dollars per 100 liter, and for downstream processing, like 10 to 20 kilogram per protein, US dollar per kilogram of protein. And for ion exchange chromatography, almost 1000 to 5000 US dollar per kilogram. As well as for stabilization of the maltodextrin, we have to use some certain stabilizer in order to prevent the drying and unfolding of the protein. So for that also we require a specific amount like we have to invest some amount. Initially, once we are set with it, then there is no need of additional investment. And for safety and toxicity and GRAS certification, it's a long time process and it's required a huge investment for the inline studies. So that's also one time investment. So once we are set with it, we can go forward for the same. And for there are certain fixed costs are there for the bioreactors, chromatography and HPLC, which need, even we have to check in every 2-3 months interval, we have to verify like the production processes. Even though in every starter culture, they will be checking whether there is no changes or there is no mutation in this particular strains. We have to verify it in a particular time interval. So that is also another important SOP for every industry. So apart from that, production scale, almost we can get almost 100 to 200 kilogram per year in the lab scale, and which can be scaled up into a larger amount according to like our yield, as well as whether we are able to properly produce it or not. Accordingly, we can scale up, because it's a air like a fed batch type fermenter. So scale upping is also very difficult task, because once we are increasing the volume, so definitely there are a lot of changes occur. So we can't scale up to a huge amount. So there are certain limitations are there. That's all. And for small scale, we can per kilogram almost 500 to 1000 US dollar per kilogram. Whereas in case of industrial scale, we can reduce, because most of the cost can be cut down, because we are producing in a larger amount. So almost 200 to 400 dollar per kilogram. That's expected cost. We cannot exactly say that it will be the cost. So it will vary with respect to other conditions.
Okay, good. So if I go by US dollars, it's like what? A dollar is 80 rupees approximately. So still pretty costly per kg.
Yes, sir.
Right?
It will be
16,000.
Like for
rupees.
example, anyway, we will be using in a smaller quantity. So
Okay.
the aim is like it is coming approximately to the even almost to 12 rupee per.
Okay, and you
equivalent to 12 rupees.
Okay, so you mentioned about stevia at one point of time, and you're talking about the boiling tea. I have never seen anyone adding stevia when we are making tea.
Stevia, actually, what I have seen, even I also tried stevia with the tea, but it will cause aftertaste, and it will be like
but that's what
it's not stable
stevia
in
is
high
for.
temperature.
But you don't add stevia while you're making the tea. You add stevia once you have poured it. You just add 2 of those and done.
Yes, yes.
So modeling can actually be considered that way.
Yes, sir.
Right? Okay. Before I go to other people, I just have one doubt, which is a bigger one. High density polyethylene is HDPE, right? Give the full form next time. FDA is
Food and Drug Administration.
Food and Drug Authority of
US FDA.
So you don't need FSSAI approval at all?
FSSAI, actually FSSAI, none of the GMO is approved. So anyway, they won't be.
So you're going to make it for US market?
FSSAI only cotton seed oil
Okay,
is
good,
there.
Good,
so
good.
that's
Thank
not
you for
food.
bringing that. In India, it is not allowed. So I feel bad now. I'm teaching you something that you might not be able to make here. Anyways, for heat stability, you will be using circular dichroism or DSC. So circular dichroism, you're going to study the structure.
Even if it's unfolding or not with respect
Okay,
to
so
temperature.
then how do you find the heat stability through CD? I have never done heat stability through CD. I've done DSC, which is differential scanning calorimeter, to like look at how the protein will denature as I am heating it or cooling it.
DSC can be used. Yes.
Okay. X-ray diffraction can be seen with respect to crystallinity,
Yes, sir.
right? So you could have mentioned that, because even ProCatalyst, you had mentioned about characterization. You didn't mention the techniques that you might use. Those are most common techniques. You would have seen some of them during the session, you just as a demo or at least visited the facility of that. Those are actually the techniques that we use. Any question for this group?
You have used the molasses as a sugar or the carbon source for your production. Basically, for a protein purification, majorly industries uses highest purest form of carbon source, so you can easily pure in downstream processes. And molasses is considered as the impure, or it contains a lot of impurities as in source. So how can you remove all kind of impurities in ion exchange chromatography?
Ah, good question. Explain the principle of ion exchange chromatography.
So I think first we can go for pre-treatment of the molasses before using it as a substrate. So that will
Okay.
reduce your impurities to a certain extent.
So, pre-treatment, that's another.
Yes, it will increase the cost, but then I think as compared to if we use synthetic media, still it will be lesser if we use
Okay,
molasses.
no, I understand that, but so you're using 2 carbon sources.
Yes, sir.
So you're thinking about dioxy growth, or you're using glycerol just as an induction to
Just
the
as
genes.
induction.
Okay, fine.
And then anyhow for the protein we are having ion exchange chromatography plus we have ultrafiltration also. So
Okay, ion exchange chromatography principle.
Sir, like based on the charge of our protein, we have to select the charge of the resin.
Okay, so monoline charge is positive or negative? Even I don't remember. If I remember correctly, it's positive.
Actually, as we are altering the amino acid sequence, definitely it will change.
It will change, right? So depending upon the charge that you'll have, you have to even estimate the charge of your protein before you go for those type of things. See, the technologies or the techniques that you all have mentioned till now, all the groups, and I'm hoping that even the last group is going to mention the same thing. They are costly, but they are lab scale. Once you have fixed it at lab scale that this is what the process would look like, for the large scale production, you don't take into account the DNA sequencings and all those things in factor. What you take into account is your source, carbon source, microbes, growth, production, operation, safety, but that's it. The whole thing that you are talking about here is done before you even go for your product development, which is a good thing. I appreciate. I really appreciate. There was one question that I did had in this particular section. Yeah, C-tab. What is C-tab?
it's a like buffer to extract DNA.
It's a pure, even for isolation of DNA, what I have seen, that's the best method to get the purest form of
Yeah, but
DNA.
what does C plus T plus A plus B stands
Cetyl
for?
trimethyl ammonium bromate.
Okay. So it's always
It's
nice.
a cationic detergent.
Yeah, it's always nice to mention those things when you're giving a presentation. Otherwise, I might not be even aware of it, right? But thank you. So no other question? Again, I will reiterate, the techno-economic analysis is not financial analysis. What you are presenting, each and every one of you have presented till now, is a financial analysis. Techno-economic analysis involves how much labor is required, how will you be putting it, what is your return on investment. There was a question from the previous group when we were, I was looking at, somewhere I had mentioned the ROI. No, I didn't mention
So
it. Sorry. The very first one, I mentioned about the ROI, the return on investment. So how much you are going to put in and how much you will get. Like, that's not just a ROI, but how much time will it take for you to break even in terms of your investment, and how much time it will take you to start making profits. Okay, prolase. The last group for the day. No, for this session, not for the day.
Hello, sir. Next slide.
Next slide? Okay.
Yes, sir. And we have chosen Candida utilis for this process.
Okay.
And the main thing which I wanted to say is not many kinds of research has been made for Candida utilis. It is still a trendy one, which is because Candida utilis is comparatively very cheaper when it is compared to other hosts. And why is it? Because it can grow on versatile compounds.
It's a eukaryote, prokaryote?
Eukaryote.
Okay.
Yes. And it grows on extremely cheaper substrate. And the main thing which is to be considered is it can grow on waste materials like industrial wastes, which is obviously good, which can make it grow on any kinds of areas which won't be environmental specific. And the other thing which I wanted to say is that waste products, sugar molasses, and those spent sulfite liquors. And the other thing is it won't produce extra byproducts like methanol, which significantly reduces the cost of downstream processing.
Okay.
It won't be producing the unwanted things which make things complex while doing the downstream process. And the main thing is ethanol in this process might act as an inhibitor, which is majorly reduced through this by the utilization of Candida utilis. And the other thing is there is a usage of plasmids and integrative vectors, which usage of integrative vectors is usually seen in this, which is why it increases the terminology which I wanted to say is high copy number expression, which accumulates a larger number of genetic material inside Candida utilis, which is the monolin producing genes, which produces larger number of monolin protein, which enhances the production obviously. And even though when compared to other hosts, which it has a slightly higher doubling time, this is incorporation of these kinds of things, which is targeting the specific genes such as rDNA or URA3 gene locus, will give us an highly efficient production, which may reach a higher cell density when it is compared to the other hosts. And when it is up to the fermentation process, it is extremely stable when it is compared to other host organisms. A research was found that even after 50 generations of non-selective growth, it has remained stable without any further major changes, which has been noted. And the common things which has been used is like centrifugation technique and precipitation, which is obviously done with the other protein things. And regarding the other processes, next slide. And the main thing which I wanted to say is it is usually done in continuous fermentation process. Why are we considering this is because for the Candida utilis, by the utilization of continuous fermentation process, it creates an optimal condition which does not make any significant changes in its density or its composition, which plays a major role in it. And the pH for its optimal growth parameters is obviously 4.5, and the temperature tolerance is at the maximum range of 35, which is actually good. And the aeration rate is 2 liters per minute, and the mode of operation which I have said is continuous mode of fermentation, which will be suitable for the scale up operation. And regarding the thermal stability, which I have mentioned in the graph which is on the right side, compared to the other organisms, Candida utilis stands high with its thermal stability, which is why it is due to its unique structure and its ability to grow in various areas. And what may be the major use of its thermal stability is we can use it in a variety of food products, which may the processes like high processing foods, which and the highly boiling point made foods, which can be a good thing actually, because it may not degrade, as it is a denaturable protein under heat. Its high thermal stability may withstand its lifetime. And regarding the monolin obtaining process, I don't want to make it say it again and again. We'll just simplify the process. As usual, we know it is an intracellular product which can be obtained through the enzymatic lysis, bead milling, and ultrasonication processes. And based on its scale-up conditions, the methods that we have for the extraction can be modified, which I mean to say is, if it's up to large scale, we can adapt fermentation process, extraction process, which can be specifically suitable for that. And regarding the contamination, Candida utilis still remains at the lowest position as possible, because the major thing which I wanted to say, it is generally recognized as safe, and Japan has approved it as harmless additive which can be added in the foods, which seems like a good thing. And the only one person who is not approved yet is the European Union, which is still under consideration. And so far, majority of the papers are only based on Pichia pastoris and the Aspergillus niger. And why we have chosen this is, this stands as a unique product, with unique host, I mean, because of its thermal stability, cost, and its substrate utilization. Then regarding its stability, for increasing its stability, certain things which is mentioned as per the research is linking the polypeptide chains, usage of disulfide bond engineering, and other genetic alterations, which has increased its stability further and further. So it seems like a very interesting host which can be further used in the future. And regarding the packaging process, since we all know that sugars are usually obtained in the powder process, we can use the methods such as lyophilization, which is the freeze drying. We can use that to obtain these things in a powdered form, which can be used in many kinds of products. And regarding the stability, I have mentioned it. And suitable additives and preservatives based on its preferences are added into it. Next slide. And here is the estimated cost production mentioned in dollars. And as I said before, it is of lower contamination. Efficient cell disruption can be done using various methods which we already know, which are discussed multiple times in this session. And it's stable expression of genetic material, which is a major positive in it, which is done using the majority, not the majority, it is used by its, integration of that plasmids, which has made its structure even more stable. And when it is compared to the others, it does not produce any kinds of endotoxin, which may act as an inhibitor while the production process. And its endotoxin production is significantly low. It is almost zero. And there is that economic analysis, which is cost per kg. We have considered three substrates which is made into an average, and in that aspects too, E. coli stands at the highest, which is in substrate cost. And Candida utilis, due to its versatile growth conditions, it can grow in, as we discussed earlier, waste products and those things, which reduces its cost in the production. And the estimated production cost, the graph is mentioned on the left side corner, which is estimated with overall upstream and downstream process, which has been actually for the Candida utilis, it is not properly mentioned, which you have made it as an approx value. When it's compared with the others, it is almost a cheaper version. When it is considered for the large scale production, it can be a beneficial and greater aspects to be considered.
Oh, thank you. Just a quick question. This is $24,500 to $30,000 per kg?
Per 10,000.
Per 10,000?
10,000 kg.
For 10,000 kg?
Yes, sir.
So it's like $3 per kg?
1 second, sir. I'll just recheck it.
I would really like to see that one, because even if I have to buy a 1 kg of sugar in Canada, I have to spend almost $3.5. So I would love to see this. I can see all of you are looking through the resource that you had used. Not a bad idea, but it's too late to use. Have you heard about perception, consumer perception, when you're trying to develop a product? The consumer perception makes a lot of impact on the choices the industries will make. So that's why, like, choosing the right host depends on, to a certain extent, onto the consumer perception, because when you're saying recombinant DNA technology or GMO, that itself becomes a big no-no. That's why European Union is still, because for European Union, anything to do with food, and if it has GMO, it's a no. It's not allowed. You cannot have it.
Sir, actually it's a correction. Actually it's
Yes,
1000
please go ahead.
per 1000 kg.
Per 1000 kg?
Yes, sir.
Even for per 1000 kg, I'm expecting what, $30 for a kg?
Yes, sir. Actually, it is mentioned as a cheaper version, and it doesn't support any approximate proof, which is just made using the substrate utilized and the approximation of the process which is used.
Okay, so this is the cost of monolin, or you're talking about the cost of Candida utilities?
Actually, it's the production cost.
It's a production cost.
Yes.
You haven't gone downstream
Yes.
yet.
Yes, sir.
Okay. Okay. Production cost. Okay.
Sorry for
because
the confusion.
the whole focus had been on the Candida utilis, right? I agree. It's an amazing host that you can use, and you're using CSTR. That's unique, and I like it. You're using bid milling, ultrasonication, enzymatic lysis. All of that will still be costing you some amount. So the final cost would be somewhere again in the same ballpark. Do you expect?
Yes, sir. Based on the production, we can
Go
optimize.
cheaper? Okay.
Based on the production.
Okay, I can understand that. But thank you. Actually, like you all have done an amazing effort, or you all have put a lot of effort in preparing this in a very short span of time. Do you have any questions for me? Yes.
You have used Candida utilis as an host, but it is like Candida utilis can cause allergic reaction in individuals, and like it can cause respiratory symptoms, skin symptoms, gastrointestinal symptoms as well. Then
Is
how
it
and why are
the
you using
product
that?
comes out of Candida utilis, or
Yeah, it is because of the protein that comes out of Candida utilis, like enolase and heat shock proteins that are harmful and can cause allergic reactions in human.
Prolase, answer the question.
Regarding the question, as I might say, it can be first when it is about consumer analysis. First, it will undergo a certain process, which will be allergen tests. And let us say if a certain product has gluten, it will be mentioned as it contains gluten. What I am trying to say is, if it does not, if it is too harmful, it might not have got generally recognized as safe, and it has got approved by Japan as harmless additives which has undergone a certain.
What about USA? They have approved it too? FDA?
USA is still under approval, sir, and the European Union has kept it as aside, as we all know that they won't approve genetically modified organisms.
So it's only Japan. And what do they use it for in Japan?
Harmless additives. Food additives.
As a food additive?
Yes, sir.
Like a probiotic?
Yes, sir. It is majorly used as food additives. It is famous in Japan for it is used as an harmless additives.
I'm going to actually go back and start looking into this. It's a very interesting thing that you have brought to the table. I appreciate it. Any other question for the group? Oh, now you have multiple questions after the first one. That's exciting.
Sir, can you go to the TA slide? This economic analysis, like cost per kg in dollars, this is I think also for a generic one. This is not for monolin, right? Is this for monolin?
It's overall production cost.
Production cost as in just the upstream, like biomass production, not downstream processing, right?
It is made up as an average cost, which is an approximation.
If it is average, don't you think it's very less? Like it's 0.5 cost like per kg in dollars for Canada Utilis?
0.275.
It is mentioned as a cheaper version, but still because of its lack of research work, this estimates still an estimation, not like a complete formation, which I would say.
Do you have a source for this information, the one which you come up with, the dollar value?
Yeah, sir. It is from there is a website for USA which calculates daily substrate costs. USA Daily, I guess so, sir. They have been mentioning.
You're saying USA Daily?
Yes, sir. I'll just mention the name precisely. If you want.
No, I like the idea if we are able to produce it that cheap, but I'm skeptical, and that's the reason why she's skeptical, and that guy is skeptical. So we need a bit more information to get out of this skepticism.
Actually, the sources which the substrate cost has been measured is Selina Wamucci. Yes, SELINAWAMUCII.
Okay.
Which is which gives the daily prices of the substrates which I have mentioned there.
Okay, so
like glucose.
This is your production cost.
Yeah.
That is what you're saying, right?
Yes.
It includes the modeling into it, but they haven't separated the modeling yet. They haven't purified the modeling yet. They haven't lyophilized the modeling yet. They haven't done all of that. This is just for till the fermenter side. Like how much it will cost you for 1,000 kgs. It will cost you around $30,000. No, they are only looking at a production. The moment the Candida utilis is kept inside the fermenter to grow, they are just looking at that particular part. They are not looking at anything else. They are not looking at the gene cloning aspect of it later. That is prior to this. So that's why I said like, you can break it into different stages, right? So it will differ in cost. Last team, would you like to counter me or counter the skepticism from the first? Oh, there's another set of questions, please.
So as you have mentioned, there are a lot of advantages of using what you call Candida utilis for production of monilin. Why do you believe that still E. coli is preferred as the host for a large scale production of monilin, not Candida utilis? They could have researched more to produce processes or techniques that can utilize Candida utilis instead of E. coli. But still E. coli is used as the most preferred host in large scale industrial production.
So what he is asking is, if Candida utilis is such a nice host, why it was never used? previously. It's not that suddenly it appeared in the world, right? Like somebody was like taking a shower, oh, Candida utilis, I should use it. It's not that, right? So why? So that's a very good question. Can you pass on the mic so that he can answer, or the group can answer? I feel like I'm some NDTV or that host, passing it from one person to another person. It's really nice. I like this session. It's very interactive.
And regarding the consideration, as she said, it has gone through various health complication based analysis, which has made it stay back in its advancements. That will be the first thing. And E. coli is mostly preferred for most of the things. E. coli is considered as a, what do you say, is become a normal thing for the production of certain materials. And why do I say we have analyzed E. coli? It has mentioned as its post-translational modifications are a little bit low. So we have gone through Saccharomyces and Pichia pastoris, and all of them had a certain complicated endotoxins that increases the cost of downstream processing. And the first point is its health complications played a significant role in its decline, which is made it still under research, and it's still under improvement, which I would say. And the next thing is E. coli is least stable under thermal stability when it is undergone in the production of monolin. And for the production, why E. coli is preferred the most? I would answer your question. It has the lowest doubling time, actually.
Okay.
So it increases the productivity yield.
So the way I understand, in the most layman way, is that you are proposing that you are going to make Tesla, and I should invest in the stocks right now and think about it later, right? It's like nobody knew that Tesla is going to come. I wish I had invested in that stock when it was starting. Okay, it was like $10 or $15 a stock. Right now, I can't afford it. But I really like the idea of Candida utilis. But as an investor, I would like to have a bit more surety in terms of the safety aspect, because any time when you're looking at investments, let's talk about a little bit from the management perspective. When you're trying to set up a startup, or you're trying to set up an industry, or when you're even trying to sell this to an industry right now, they will not take it because it's not there yet in terms of the approvals from the safety perspective. One question that I have for all the groups, what you all had mentioned is setting up a new process, right? Industries, some of the industries, let's say if it is Cargill or my favorite that I can take as an example in India, Biocon. If let's say Biocon already has a fermentation facility, it already has everything set up, will you be able to utilize their facility as is, or you have to make some modification? Or do you need something else? That adds cost, right? Do you think production of monolin will require extensive investment into technologies that are not commonly used in a fermentation industry, or like a high product development industry, or what would be the thing? Can I use the setup that is there right now? Then I don't have to make any modifications, right? So I can go to the industry and say that, hey, here is the product that has a really high value. Would you be interested in looking at putting it through your own pilot scale studies and see if it works? If it works, they can have it as a product. They can have a different setups already existing for this. You need a fermenter. You need all the lab facilities. So technically, any industry that already has it, they can do it. But you need expertise in this. from the R&D perspective and also the processing side. But I like the idea of Candida utilis. Thank you for bringing this to our discussion today. Any question for the group? No? So, in that case, we are done with our case study 1. We are going to start our case study 2 later around 2:30 p.m. Okay? Thank you. Good afternoon. Welcome back. I hope you had a nice lunch and you have some energy to put back into our conversation that we have been having. So, we are going to start the second session for today, and this will be the last session for this GIAN course. So, this is on the case study 2. So, for the case study 2, it was about developing a microbial protein, a protein that can be produced by the microbe, or which is actually produced by the microbe and is utilized in the industry on a wider scale. So, the one which I gave you as an example, or not as an example, I wanted you to work on would be alpha amylase. So, alpha amylase is produced by the microbes The idea was you are going to use the techniques that we had discussed as part of the synthetic biology to impart properties into alpha amylase that will make it much more viable. Alpha amylase is not a new enzyme. It is widely used in the chemical industry. It is widely used in the pharmaceutical industry. It's widely used in the food industry. So, it has been in the market for a very, very long time, but there are issues which are still existing with respect to its production, with respect to its quality to a certain extent. I will not say to production. Production has been always been optimized for a majority of these products, but quality does matter to a certain extent. And our intent was to come up with a newer solution using precision fermentation. Now, the definition of precision fermentation is where you are using something which requires recombinant DNA technologies. You are producing it in a microbe on a larger scale, making it much more viable, correct? So, I will start the same process the way we started having the conversation in the first session. So, we will start with the group or the person who submitted the case study first. Mr. Rajesh?
Yeah, I will start.
So, what we have for the case study 2 for Mr. Rajesh is again the same company, Swiss Shift. You are not changing the company name? Okay. So, it is a second product from your own company. Cool. I like it. So, you have diversified, which is a very, very good thing. You know, like the idea of alpha amylase, like many people have obtained alpha amylase from soil. It's like, it's an enzyme which is produced by the microbes when they are in the worst condition possible. Like if they need to just survive somehow, like they're clinging to death, hanging from the cliff, like that type of situation exists for them. That's when they start producing this enzyme, and it's actually very beneficial for them, for their survival, and that's the only reason why we are looking at it. Because it's a prominent enzyme, it's widely used, there has to be something new that we can add to our conversation or through our conversation. Do you have any questions with respect to the case study 2, like how you would have approached, or no, not case study 2, case study 1? Till the time they fix the technical issue, we can go there. If you don't have any question, Mr. Rajesh, if you can explain what exactly you did and how you approached it.
Good afternoon, one and all. So, I am with my second case study, that is developing a microbial protein, alpha amylase. Okay, so where we are coming with the same team, I mean Sweet Shift. So here I went on coming to the procedure regarding the conversion, making this. So then starch is the one of the best feedstock for this one. So the country like India, we are having huge amount of starch as per the reports or something. So 17% of world's agro residue is having there in India. So I read with the different papers, they are using every residue. So like potato or like wheat, barley, corn. So and whatever the residue is there, they made different. I mean they used all these agricultural residue feedstock, and they tried by using these bacteria one I mentioned, Bacillus, this one. So and they changed the gene modifications with respect to that one and made. Recently I read one paper in 2024, I think that one I read about that one. So where I selected these here bacteria which can give the rapid production, or for the enzyme production it will be helpful, like that, that's why I selected. And the gene modification and where the gene cloning, where these the parameters are there that I am presenting. So giving the carbon nitrogen sources, and here the temperature we are
So this is for your
for this
the fermenter.
Ferment.
Okay.
Yeah, for the fermenter, the temperature source 37 degrees, and the speed also mentioned. So some mentioned 250, 260 like that. So as in medium like that, I went with 180 to 220 RPM.
So this is something that you decided upon.
Yeah, I decided. So they went for 250 to 150 to 250 like that. So I opted it like this is, and the pH value 6.5 to 7.5, and the dissolved oxygen 30 to 50%, and the carbon sources will be the glucose, and the nitrogen will be the ammonium salt. here for this, if you went with the exterior tank, so this will be the best one. So then I selected for the batch fed batch reactor. Even people did with this reactor. So and where the main reason is we can add the nutrients as in this one in the substrate, and here the purification, I mean the extraction process includes the intercellular setup, where the cell lysis, protein extraction, purification, and the concentration, this can be there, through which the alpha amylase can be obtained. And coming to the techno-economic analysis, where so the same I mean previously we discussed about in the plant protein production. So there the same methodology I followed. The raw materials, and the where the cost bacteria and the cost of starch and the enzyme modification. Here the enzyme modification cost reduced because people did on this when compared with that one. So that's why I reduced some cost in this, and the energy cost for the aeration and the stirring and heating are there, and the purification where the filtering and centrifuge, and the capital cost, which includes the reactor and accessories and space infrastructure, and the labor cost adds the same like skilled and semi-skilled. So here the thing is the total cost budget that I proposed. Oh, sorry, the it's a blunder. Okay.
I didn't mean to disturb you.
Yeah, sorry. And the total cost which is actually, if you write a proposal with respect to small one batch like proposal, it can cost of 10 lakh. Where here for India, the country like India, this as I said, we have huge amount of the agricultural based residues which can be available, through which we can use this generate this alpha amylase protein. So and the potential applications are also much more when compared with like these food beverages and pharmaceutical and the bio type detergents and these animal feed, some mentioned, but like that. So here so that's why so when compared with the others, so alpha amylase production is done huge in India, so it has a fruitful one. So anyone can do the change in the gene and they can run it.
Okay.
Yeah. And sorry for the mistake. That is.
That's okay. That's okay. Even that was there in the previous.
And
one.
This was preliminary survey that I did with respect to some papers.
No, I'm glad that at least you had an opportunity to go do the survey.
Yeah.
Right. So at least you got to know that there are certain things like monolin and alpha amylase that you can look at.
Yeah.
As an entrepreneur yourself, you should have broaden your scope. Cool. One thing that I would like to bring to your notice, like alpha amylase, as I said, is produced by the microorganism in a worst case scenarios, like in the worst case situations of it. So the question to you would be, so when we are looking at the feedstock, we had conversation a couple of days back about the paralis and all the burning of the paralis in Haryana and Punjab. Do you think setting up a small scale industry where you can actually collect all of these feedstocks, break it down, because you have to do some sort of pre-treatment to the feedstock. You cannot use the feedstock as is, right? I cannot use the stocks of the plant
directly
into the fermentation system. So what about the pre-treatment? Because I haven't heard about pre-treatment from any of the groups till now?
So basically from 2019, I am working on the agro-residue management.
Okay.
So through which I started my startup also. So even, so the thing is, so collecting the residue in a clear way when I am going to it, collecting the residue from the farmers is a big challenge. Because if you want to collect, you have to start this one in before their, before their crop growing time. So before in the months of June, July itself, we have to talk with them such that you keep your residue aside and like that. So then what we will do means, and we have to monitor their production, and before they are setting their harvesting time, we have to collect the residue and we have to keep them in a host zone like that. I mean, we'll call it as an buffer zone or like that, which is of 50 kilometers.
So you are saying you are going to collect the residue before the harvest?
After the harvest. Okay. After the harvest, before itself, we have to go and talk with them that don't throw it out.
You're making the arrangements with the farmers.
Okay. Yeah, arrangement for the farmers. And we have to collect them and have to keep them in then stock or go down, which is in like a mandal, right? Like mandal type. That is 50 kilometers max, 25 to 50 kilometers of that one. So there we can set up a small go down. Okay, so that is the stock point. And with respect to the stock, we have to make one arrangement. So to such that the stocks are within a range like that. So such that if you keep with respect to a mandal, so usually the mandal will be of this much size only, between 50 kilometers we can get that, you can keep a stock point. So then after this, then the pre-treatment process, there you can establish your warehouse through which you can run it. So the same model we proposed in our district, and even I am following the same model for my startup.
Can I add to that?
Yeah, you can.
So Food Corporation of India has godowns, has the space, has the facility. Why not utilize what you already have?
So
why do you have to build a different warehouse?
So actually food processing, this one is there, godowns. Food Corporation of India godowns. So there will be not, if you see for a district, they won't keep 1 or 2 for a district. As for my district, I am from my district, district. My district from Andhra Pradesh. So my district has 3 itself. Okay, so which are in that in this range only, 50 like that, and they will think and they will charge actually for this.
No, if the government is also interested in that, I don't think government will like to bill itself for doing something beneficial for the farmers. I'm just proposing. I'm just giving you an idea. You can initiate a conversation with the MLA and the MP of that area and have a conversation about FCI. But coming back to your thing. Any questions for him? Okay, if they don't have any questions,
thank you.
I have only one single question. You're going with the same process of cell lysis, protein extraction, purification. My question to you is, why do you want to purify alpha amylase to a pharmaceutical grade level? Why can't we just use it or keep it up to the food grade level? Because food grade level is not that high, you don't need that much of purification, and you might be able to make it cheap as compared to the pharmaceutical grade.
Based on the, I don't know that answer, but I am saying as
no, I'm just suggesting. So if that is a possibility, let's say, will you be interested in doing that, like reaching only up to the food grade level quality?
Obviously, food grade will give the more because.
Will it give you a bigger market as compared to the pharmaceutical grade?
Currently, pharmaceutical will get more margin. Suppose in small medicine also they will take more.
You're talking about money. Nice. You're all thinking about the dollar. Sorry, not the dollar. The rupee sign.
Indian rupee.
Yes. Good. I appreciate that.
Thank you.
Thank you. I like that entrepreneurship in you. But if you're really interested in these type of businesses, you seriously need to learn a little bit more biotechnology. So gene cloning and how you're doing the recombinant DNA technology, or at least hire somebody from the biotechnology group.
Yes, sure, sir. Actually, I missed the same for this one. Even I applied for the BRAC, okay, in 2020 itself. So there I got to know that I missed in 0.14. So I never heard about that much after 0.14 values.
Good.
So 0.14 I missed the project. So hope
That's
next
fine,
future
that's
I can
fine.
join Blend.
No, but see, there are the reasoning is that when you're looking at feedstocks just like that, for example, in the previous session when we were having the conversation, we talked about the monoline protein if it is added in the food product itself, and then we can definitely do pasteurizations and things like that. I think it was the third group, Bluff, right, that you suggested that. But when you are doing, like, because for the pasteurization, you're going to use heat exchangers. If you have protein in it, it might change its structure, and that might lead to flocculation, and your efficiency of your heat exchanger will reduce. So what will you do? It can even denature your protein, but let's say if you are able to keep it in the starch, maintain its structure and everything, but there might be a situation where it will interact with other components of the food, because you don't know, like, how the protein and starch relationship, right? So when you heat them at a very high temperature, what sort of reaction happens? Maillard reaction, right? I'm sorry? It's more than 100 degrees Celsius? Yes. Okay. Okay, good. Any questions for Rajesh? There is a question at the back, please.
So basically, what are the gene modifications that have been considered for this particular experiment? You know, like your thoughts, like there is some kind of idea, right?
No, at least whatever you have studied in this particular course, you could tell that. If you want to, like, you want time to go back to your notes. See, I think, see, for alpha amylase, you would be looking at the same. You take the microbe that actually produces it. You don't have to, sometimes you don't even have to do the gene modification for it. You take the microbe that produces it, but you might take the genetic DNA and insert mutations so that you might get the alpha amylase with the specific properties of interest. That is a possibility. That would be the approach that I would like to take, because that would be cheaper for me, rather than taking out the gene of alpha amylase and then growing into something else.
Yes,
But I will choose the organism that actually produces it. Just requires the enough carbon source, but I will definitely like to initiate some sort of mutation in it to get the enzyme of my interest. Heat stability is a very important factor with respect to alpha amylases. The alpha amylase which is widely available in the market, they are not very heat stable. So that sort of mutation is something that I'm expecting.
But sir, there are like vectors now available, like you can say like PUB 110,
Yes.
which provide like, which also contains like initiation signal, like ME signal that can promote more production of the required protein of interest, which is
yes.
alpha amylase.
See,
So
production itself is not an issue. Like, okay, if you want to produce a lot, it's fine.
yes, sir.
If that's the only requirement. But if I want you to produce it, but I want you to have a specific property attached to it, if it doesn't have that property, then it's not valuable to me. Right? Enzymes, because by virtue of their functionality, right, by virtue of their functionality are important. If they are not functional, they are useless. They have to have a specific function to it, right? Amylase has to cut. But if it is not heat stable, then it might not be a viable option for me if my application requires it to be heat stable. I'm just putting out an answer for him. This is not my answer alone. I'm just putting up an idea there. Any other question? No? Then in that case, we will go to the team procatalysts. Should I change the slide?
Yes. So good afternoon, one and all present here. The case study 2 is developing a microbial protein alpha amylase. So alpha amylase is a starch hydrolysing enzyme which breaks down alpha-1,4 glycosidic bonds. Alpha amylase is used in various industries like food industries to convert starch into sugars. They are also used in textile industries, detergent industries. So it concludes that alpha amylase is a very important industrial enzyme. And for producing the alpha amylase, we have selected a bacteria, namely Bacillus alkalophilus. And the reason for choosing this bacteria, basically, it produces a stable amylase protein which stands up to 55 degrees Celsius, and it is an alkaline alpha amylase. And the vector the host we are choosing is again the Bacillus subtilis, WB600 strain. And a reason for choosing this strain is basically in this strain there is a knockout of 6 genes, and those genes are basically responsible for producing the protease enzyme. And the protease enzyme basically degrade the recombinant protein. And in this WB600 strain, those genes were knocked out. So we can prevent our protein from degradation, actually. So the process goes like that. And first we have identified the gene. The gene sequence was given there. It was not the full sequence. I have only taken a small part just to prevent the space. And after that, vector and host selection was done. Vector was taken as the PMA5, which is an shuttle vector. It can replicate itself both in E. coli as well as in subtilis. And the host chosen is Bacillus subtilis WB600 strain. After that, the primer was synthesized for the specific gene sequence. And in that primer, there are restriction enzyme coding sequences also, because we want our gene to be specifically ligate to our vector. So we have incorporated 2 restriction enzymes, the BAM1 and SAL1, BAMH1 and SAL1 gene into the sequence. And then it was amplified using the PCR through at standard procedures. And after that, both the gene and vector was digested with the restriction enzymes, the same restriction enzymes. As it contained the sequence, so it will ligate to it only. Then ligation reaction happens, and then selection was done to differentiate between transformants versus non-transformants. It was done using 100 microgram kanamycin per ml and 1% starch. So 1% starch is used because if our transformed cell has amylase gene into it, it will basically hydrolyze it. And we can confirm that these colonies are having the amylase gene and these colonies are not. So by picking up the transformed colonies, we basically cultured and gone for the pilot scale fermentation process. Change the slide, please. So this is the vector design. As you can see, our alkaline alpha amylase is between the 2 restriction enzyme sites. We have incorporated the sticky ends and it was ligated into the vector. And the right hand side figure shows it is basically a SDS phase diagram in which जो arrow है वो basically 56 kilodalton protein in which confirms that our crude has the alpha amylase into it. So it covers the upstream process, and the downstream processing will be explained by my partner Vishal. Thank you.
So after incorporation into Bacillus subtilis, so the mode of fermentation that we have used here is solid state fermentation instead of submerged fermentation. The reason being it will have low moisture content, better product recovery, low water output, and lack of foam built up. The carbon source we have used is the substrate that we have used is wheat bran, which is an agricultural waste. And then the nitrogen source we have used ammonium sulfate, inorganic source. In the study, we have found that the pH, the optimized pH is 7.1 for the production of alpha amylase using the Bacillus subtilis, and temperature is around 37 to 40, but on a study we found that the highest amount of alpha amylase production is around 40 degrees Celsius. And the bioreactor that we have used here is tray bioreactor. Now, sir, can you change the slide? Now the downstream processes include extraction, filtration and centrifugation, precipitation, dialysis, purification. concentration, enzyme activity assay, and storage. So for the extraction purpose, it is basically used to recover the enzyme from fermented substrate by using an appropriate extraction buffer, example phosphate buffer or trisacil to the fermented material. A solid to liquid ratio is maintained, that 1:5 to 1:10 to optimize enzyme recovery. Okay, so after that, after getting the alpha amylase by shaking the mixture to facilitate the release, we can use filtration and centrifugation. Obviously, filtration, we use coarse filtration to remove the large particles, then centrifuge it to clarify the supernatant. After getting the supernatant, for the precipitation and purification process, precipitation is done. We use the crude enzyme for 0 to 30% cut. We will use the precipitation dialysis process, which include the like 0 to 30%. We will add 268 mg if into 10, that is 2.68 gram of ammonium sulfate. Then the stir the mixture for 30 minutes. The solution is centrifuged, pellet is taken out, supernatant is kept aside, and the pellet is again resuspended in in the phosphate buffer. The supernatant that is kept is again used for 30 to 70% cut, and again ammonium sulfate is used, and again the dialysis is performed. After this, the purification, the data that we get, so can you go to the previous slide? The data that we get is protein concentration is 0.17 mg per ml. The extract activity was 2.243 micromole per ml per minute, and the specific activity was 13.19 micromole per minute per mg for 48 hours of extract. Now, for the purification, enzyme activity assay, we can use the DNS method for enzyme activity assay, and for storage or the SDS page also. For storage purposes, we can use stabilizers like glycerol or sorbitol. For short term use, we can store into 4 degrees Celsius, or for the long term, we can use lyophilization or like freezing.
So you are generating a powder?
Yes. And the cost economic analysis will be done by my friend.
Good afternoon. So the first step will be the clone of the amylase gene into the host organism Bacillus subtilis WB600 strain. So we found out the approximate the value of the for the gene editing was $5,000 to $10,000. So the second step will be the media formulation, where we are using the carbon source as the wheat bran and nitrogen source. So we found that for the 100 liter of the batch culture, we have to spend almost $12,000 to $200 for the 100 liter of batch. And the inoculum preparation for the seed fermenters was found to be $500 to $1,000 for the seed fermenter preparation. Since we are using the tray bioreactor, it was found that the tray bioreactor will range from the $10,000 to $30,000 as per our need. For the as we know that the cheap one is also has the manual control, as well as the expensive one have automated manual controls. So the fermentation process includes the temperature control, pH control, and agitation control by including all the expenses with the labor and electricity energy consumption. We have concluded the approximate value up to $2,000 to $5,000 for the batch for the fermentation. So the cell harvesting comes with to separate the biomass from the fermentation broth was found to be by the centrifugation. By using the agitation speed and energy consumption, we found to be $1,000 to $2,000 per batch. And the cell lysis, which was done by the high pressure homogenization. So we have to find out the most proficient one, which was found to be up to $1,500 for the instrumentation, which was the one-time cost. So the purification, as Vishal said, the purification includes the ammonium sulfate precipitation and dialysis, which is quite expensive, which concluded up to 5,000 to 8,000 approximately dollars for the per batch volume. For the quality assurance, we are using UV visible spectrophotometer, which was we have to spend one-time cost for that spectrophotometer, which was found to be around in between 750 to 1,500 for the quality assurance. And ultra filtration for the 4 high grade, we have to do the ultra filtration to remove the salts and concentrate the proteins. It was found to be $1,500 to $2,500 per batch. And drying and packaging, which was done by freeze drying and lyophilization, or the lyophilization in the powder form for the storage for the convenience of the transport and storage condition, it was found to be $1,000 to $3,000 per batch. So we estimated up to our one-time capital with including the labor, energy cost and everything. We was found to be our yield was up to approximately 10 to 50 grams per liter. So we have to find out the exact value so we can sell out. It was found to be $1,000 to $5,000 $1,000 to $5,000 per kilogram of the purified alpha amylase for the food grade quality. Thank you.
You said food grade quality.
Yes, sir.
After dialysis and all.
For we are doing the ultra filtration for that.
Ultra filtration and dialysis, you get food grade quality. You are getting the best grade quality actually.
Pharmaceutical, sorry. Amylase, sir.
Okay. Any question for the group? Don't be hesitant. I think you all have had like a meeting. You don't ask me, I don't ask you. Okay, let me ask them. Okay, why solid state fermentation?
It will lead to low cost because downstream processing is a little bit easier. Low moisture content we can maintain in the submerged will have high moisture content.
And the pH of 7.1, if I may just wait here for a second, you said pH 7.1, 6.5 to 7.5.
Actually, I referred to a paper where there was a study being done for beta subtilis, and it was measured for the pH optimization and temperature optimization, and they got the temperature 7.1 around 7 and temperature around 40 degrees Celsius.
Okay. Not a bad idea. This would be solid state fermentation is cheaper, it's easy to maintain, easy to clean. But the question would be, you said in your, I'll say, economic analysis, not the techno part, economic analysis, you are spending $10,000 to $30,000 for this large scale fermentation. That's a one-time capital expense. Is alpha amylase the only product that you are going to have out of that fermentation system? Year after year, day after day, month after month, sorry, day after day, hour after hour, day after day, or week after week? Or you're planning to produce something else also? Because $30,000 if I have to spend, I would like it to be versatile enough.
Sir, we're actually planning to use 100 liter batch. It maybe comes in a pilot scale. So here we can do multiple product facilities where we can produce multiple products. Like if you see the process, it is actually same for the previous case study also. It is also same for this case study also. So this, and I have seen the many industries developing this scale for facility for testing the many products before going to production scale. So in small changes we can use it for a multiple product facility. Yes.
See, 100 liter is a pilot scale.
It's pilot scale, yes.
The reason I am asking this question is because I personally didn't teach you the scale up of solid state fermenters. How will you do it if you have to scale it up? What is the basis of scale up in this case?
Sir,
we are not looking at the tip speed anymore. We are not looking at the aeration or we are looking at the aeration?
Sir, if you talk about specifically fermenter for solid state, we can introduce to multiple tray or in a multiple sheets in between to increase the surface area.
Okay.
So this can be introduced in specifically fermenter.
You have seen like that? You have read about it?
Yes, I have read about that we can give the extra sheets or extra trays to increase the specific surface area for the cell growth.
Okay. Do you have anything else to add?
Just this stacking we can do, like one above the other, and maintaining a 10 to 15 centimeter of space. It will be enough.
And each and every tray is still porous in nature, so that the media can drip in.
So what?
How do we add the media?
So we can provide a media by dripping itself, or we can supply it through the specific tray.
Okay.
Yes.
Scale up would be interesting thing for me.
Scale
I
up
do like the idea of having multiple trays. You are basically using the same facility. You are increasing that overall, but the volume does not change, right? So you are not changing the volume. You can just changing the spacing between the microbes. I don't know how productive or how efficient that process would be. That's the only concern that I might have. But I like the idea of using solid state fermentation for this. I have read too some papers where they have actually used solid state fermentation for alpha amylase production. So I'm happy that you were able to find some one of those papers to go ahead with. Anybody has any questions for the group? No? So it's like 1,000 to 5,000 per kilogram, 1,000 grams. So that means $1 gram. Not bad. It's cheap. It's actually very cheap. Good. Thank you. Bluff. Your turn. Bluff is.
Good afternoon, everyone. This is your team Bluff from the morning session. And for the second session, we'll be explaining how we'll be basically the entire bioprocess design for the production of alpha amylase. So I'll be handing over it over.
I like the idea. You initiate it and you just transfer it.
So, moving to the alpha amylase, as we know, alpha amylase is used in various industries, especially food industry, like the nutraceutical industry for controlled delivery of nutrient, like different bioactive compounds, and in food industry in such a way that in breweries, like bakery industries, and other fruit and vegetable processing industry, we are using alpha amylase. And in case of alpha amylase, as you know, like for example, if we are talking about the bakery industries, we have, or in brewery industry, it should have the ability to withstand the temperature. But the bakery industry, the concern is that at a particular temperature, it should be inactivated. That's a major constraint. So accordingly, we have to modify the enzyme, and even naturally, we can see that there are various types of alpha amylase as extracted without genetic engineering from various sources like fungal amylase, and for barley malt they are extracting alpha amylase, and like Bacillus subtilis and Bacillus cereus species, which also produces naturally alpha amylase without even genetic modification, and some other organism also producing. So why we are going for this DNA technology specifically for this extraction and for improving the yield in such a way that we are using Bacillus acidocola. So this is organism which produces heat stable alpha amylase, and our aim is to focus on brewery industries because we are giving a higher temperature, and for the like as we know, alpha amylase is also known as liquefying enzyme, beta amylase is saccharifying enzyme. So the liquefaction is required in breweries to produce like initially we have to produce the specific amount of glucose for the utilization of yeast. So first of all, we have to introduce this enzyme instead of instead of introducing the glucose source as well. So from malt we have to convert it into glucose. So for that purpose, we are like our idea is to produce alpha amylase for the same. So Bacillus acidicola, and we are usually it is present in soil. So we are isolating different cultures and different strains will be there. So from that we will be isolating the strains which produces higher amount of alpha amylase and which can grow in like a different variable conditions, and which can produce a definite proportion of alpha amylase. And after that extraction of the gene, we will be identifying the gene and design the primer and like primer for the same, and next followed by the PCR amplification, then cloning into vectors, and we will be specifically selecting the vector for E. coli, that is PET, that 28A plus, the specific vector we will be selecting, and after that it is introduced into E. coli. As we specifically selected E. coli in such a way that even in previous slides you might have seen that E. coli is intracellular production of alpha amylase, even though the production is quite high and this generation time of E. coli is quite lower, so it can produce in a shorter period of time. And so after that, the selecting in the vector and the vector preparation, so digest the plasmid with a restriction enzyme and phosphorylate the vector and to prevent the self ligation. It's followed by the ligation process, ligase the PCR amplification gene into the linearize the plasmid using DNA ligases enzyme, then prepare chemically competent E. coli, then add the ligation product into the component cells and heat shock or electroporate. Like that's the method we are used for transfer the genes, this particular gene. There are different types of like gene transfer mechanism are there. So heat shock is the best method, as we know, like even different types of methods we are using for heat shock. So impulsive electric field can be used for the heat shock in order to create the pores, and like reversible pore formation and transfer the gene. So we will be limiting the electricity for the same. So after that culturing, after that the gene expression we have to see like whether it's producing the particular, it's producing the alpha amylase or not. After that test the activity of the purified enzyme to confirm its functionality, whether it's shown the heat stability. Even sometimes it may chance of even already we are using the electric shock, sometimes mutation may happen, we can't say, but it may create some changes in the protein production. So even though we have to check whether it's showing the specific properties or not. So after that the gene expression. We will be checking for the same. So after confirmation we will be selecting the particular strain for the so there is a mistake like by editing is the alpha amylase production, not monolith. Okay, title. So title. So alpha amylase production. So so low cost. We'll be using a low cost carbon sources like molasses can be used. But even though like even purification is another matter, so we'll be checking for the same. So the nitrogen source which consists of peptones and ammonium salt. And even though even mostly alpha amylase for food grade it does not require that much purity, but even though like purity is required, but for example brewing and all anyway it's going for distillation, so purity does not create much issue. It's not a big deal in case of brewery industries, but for pharmaceutical grade it is important. And next one is a salt which is commonly supplied sodium chloride and magnesium sulfate, and substrate for the induction. Specifically we will be using lactose based system because it's instead of IPTG. And fermentation conditions, specifically there are like in initial case we will be following the growth temperature. For the temperature is almost 37 degrees Celsius, and for in case of during induction period it is 30 degrees Celsius. And this IPTG is nothing but isopropyl beta D thiol acetopyranose, and dissolved oxygen is also required, that is almost 300 to 500 20% keep about 20% using, like when dissolved oxygen we have to keep about 20% using air sparging, and agitation is almost 300 to 500 rpm we have to provide, and during induction stage it is 200 to 500 rpm based on the oxygen demand. And the type of fermentation we are following, like either for small scale production batch fermentation, and for large scale production fed batch fermentation. And during harvesting induced OD, like we will be checking for the OD value that should be at 600 nanometer, so it should be in the range of 0.6 to 0.8. In that condition we will be harvesting it. So in case of downstream processing, so as I already mentioned, if it is for, depends upon, once we are focusing on the brewerage purpose or brewery grade, for in beverage industries there is no need of higher purity. So but in generally we have given the procedure for centrifugations at 8,000 to 1,000 rpm, and for almost 15 to 20 minutes. So we'll be separating the pellets of E. coli. Then cell lysis has to be done because it's an intracellular enzyme, so we have to break down the cell. So that is HCl buffer, which is commonly used, and lysozyme, and the sonication, ultrasonication can be also done for the cell lysis for the easier separations of the in cellular components, and we can isolate this protein. We have to degrade the proteins. So meanwhile we have to assure that the alpha amylase should be retained, and after that followed by centrifugation, then purification, and we are following affinity chromatography, that is nickel NTA, that is nickel nitrotriacetic acid, which is used as a resin, and for histamine tagged alpha amylase, and wash the column with low imidazole buffer and dilute with high imidazole concentration. If the enzyme lack tags, employ ammonium sulfate precipitation and ion exchange chromatography, and followed by the purification stage. And for that, size exclusion chromatography is commonly used in order to remove the aggregates or other impurities. And for stabilization, so for example, if you want to keep the enzyme for a long time, we have to bind it with calcium plus ion to stabilize it, and followed by the lyophilization and formulation into the buffer. For example, we have to keep it, we have to stabilize, the pH should be maintained nearly 6 to 7. And one more thing I forgotten to add in that, E. coli, which produces some protease enzyme, so we have to knock out the gene corresponding to the same. That's it. Thank you. So techno-economic analysis. So we haven't even gone through in details about the techno-economic analysis, but approximate cost for almost we calculated $145 per kg of enzyme, because according to the beverage grade we don't need to purify in details, but if you are going for chromatography and all, then definitely we have to this cost should be increased up to a higher level.
Okay.
But if you are eliminating that stage, we can reduce the cost.
Okay.
Thank you.
Thank you. Now if I just want to go back, constip, liquor, molasses or whey as a primary source. Okay. Do you know anybody who can sell me whey for $5 a kg? If you know, please give me the address.
In India there is.
This is Indian, or you're estimating in dollars?
Generally, we are saying whey is not commonly used in most of the industries. As per dairy industries, even most of the industries, they are just what I understood is, even whoever working in dairy industry, whey is not at all utilized. Only Amul is only producing whey proteins from whey, and even it's too costly. That's why the whey proteins cost too much. That's why most of the industries.
So where are you getting those whey protein isolates?
Even in India, only Amul is producing, as per my understanding, because most of the industry, they are just recycling it. Most of the dairy industry in India, they are not utilizing.
Okay.
So definitely, instead of just going for their
you know that it's not like that they have a, oh, here is the milk, here is the whey, and they can give you the whey in hand. It's like it's drained out
They are like
in the
even
drain.
draining only.
So you will be collecting it from the drain.
No, sir. We can directly collect from them. If, for example, otherwise they have to go for their secondary treatment. They have to
Pre-treatment of your product, pre-treatment of your feed stock is always important before you go into your production.
Because in dairy industry anyway, after whey will be collected from dairy industry. Even after pasteurizing, they will be going for this coagulation. So definitely it's pasteurized whey. So no worries, you can take it.
I like the confidence, but I would really like you to pass the FSSAI board then. With that comment, you just mentioned that it's already pasteurized. Why the hell I read this? No, you don't need to. You have to. Any question for the group? Oh, you have multiple questions. Good. Which part? The recombinant DNA part.
Actually, I want to know how did you incorporated the his tag to alpha amylase?
Wow.
You have not mentioned here any gene sequence or anything.
Oh, you're talking about this one.
Next slide, sir, I guess.
The his tagged alpha amylase. That's not in the recombinant DNA part. That was basically for the affinity chromatography.
Yeah, but sir, they have to incorporate previously so that they can use in the affinity chromatography.
How did you do that?
How did you do that?
You have to have the mic to answer that question. It's okay. It's okay. If you don't know the answer, just say you don't know the answer. You can look into it and you can get back to it.
I don't know exactly the gene editing, how it's done, but even like 6 histine, corresponding gene corresponding to 6 histine can be introduced along with the primer.
Yeah.
So we can tag it.
Okay, good.
So I didn't add it exactly in this slide because it's already we discussed regarding this in the previous thing.
Okay.
So
No issues. Any other question? Yep. Procatalysts have a lot of questions because they're a competitor actually using the same technology. Good. I like the competition.
Sir, can you please go to the costing slide?
The cost? $5, $10, $20 for?
Sir, for purification, they use a 2 chromatography system. One is affinity chromatography and another is size exclusive chromatography. And using both system in large scale, using multiple solvent, multiple things, how can you manage in $60 only?
Using the NIT facility. That's the cheapest way you can do, right? Because if you have a technology business incubator here, you get free access.
But still, sir, the price of solvent, electricity, it's...
Yeah, you convert 60 multiplied by 80.
5,000 rupees.
Right, because you are using US dollars, right? So you're
only 5,000 rupees.
Oh, yeah. Then you hire one of the master students to do the project, then it becomes free. It's a very good question. To be honest, this is wrong. I think you just did like, okay, you know what? We have to do this. 5, 25, 25, 30, 60. It's just random values. Like the numbers that you have chosen, 5, 25, 10, 15, 60, 20, 10, those are random values. Like it will come to your mind. If I was in your point, I would say, okay, 500, maybe 1,200, cell lysis, maybe 800. You can do that way.
They are bluffing, sir.
But I know, I know. That's the bluff, right?
Yes, sir,
because
that's
the team
the bluff.
Team bluff is trying to bluff us out. I'm not happy. I'm not happy. With your techno-economic analysis, you get a 0 star. Just for this. Okay, I do have a question. I know this is the last session, so we are having a nice time, but I would definitely love to ask you a question. When you say 200 to 500 RPM based oxygen demand, like RPM for the agitation purposes, what is the size of the bioreactor you're talking about? You need the mic to answer that. Like for the data that you have here is for 100 liters? It's a value that you are estimating or the source that you used actually had these values?
Sir, for 100 to 120 liters only, we have referred the papers. In that they have mentioned like for E. coli, it's not specifically for the production of alpha amylase.
Okay, just for the cell growth.
Cell growth.
So you're taking the gene from the bacillus, introducing it to E. coli, so E. coli becomes your host. Okay, and then you use the DBTL, which is good. I liked that idea. And so this is for 100 to 110 liters.
Yes, sir.
I still feel 200 to 500 RPM is pretty high. I'm trying to imagine. Is there a fan here? No, we don't have a fan here. What would be the RPM for a fan? Somewhere 100 to 200 RPMs?
150.
So if I take that and put it inside a 100 liter, I don't think my E. coli will last any. They will be like flying everywhere. No, I'm just curious. I'm just curious. This 100 to 200 RPM seems a bit higher. I might be totally wrong. You are using the sources. I don't know. So because you have to mention the resources.
Actually, we don't have practical experience while doing this, so we have only the papers which is available.
No, I agree to that, but it would be nice to have, whenever you're making this type of presentation, it's always nice to have the sources so that we can go back to them and just see if it makes any sense. But I like what you did. Thank you.
Thank you, sir.
Okay, the last group. No name, or you are the group of Mr. Aswath Vishnu? What's the name?
Sir, our group name is Prolase.
So you are still Prolase, right?
Yes, sir. Pro means
PROLAYSE.
Combination of protein and alpha amylase.
I like the name, but I'm just saying, like, it appeared like this on my screen.
Okay, sir. Yes, sir, we can start.
Okay, proprietor. We can mention this.
Sir.
Okay, good. So production of alpha amylase from Bacillus licheniformis using directed evolution. Nice, good.
So here I have used Bacillus licheniformis. Why means it is a commonly used microorganism for enzyme production, and like it is a natural producer for alpha amylase, highly suitable for hosts, and then it exhibits rapid growth under optimal conditions like 37 degrees Celsius and under pH of 7.3, and then it can reach high cell density level, density level at QSL. It is for cost effectiveness, and then one key advantage is there. It is like Bacillus licheniformis is there. It is the ability to secrete alpha amylase in the culture media. So it reduces the downstream process cost.
So it's extracellular.
Extracellular. It is extracellular. So it reduces the downstream cost. And then coming back to the recombinant bacteria, like we have taken the strain like CBB0302. It is a strain, and then we have taken the vector PHY AML, and then we just integrated with recombinant technology, and then some preparation processes that like carbon source like sugar, and then nitrogen source like peptone, and then yeast extraction, and then phosphate. Phosphate group is there, and then trace element is also there. Zinc, iron, magnesium, it is there, and then now comes under to the there is no direct evolution. Morning we have updated the presentation, sir.
This is the only one that we received.
Morning we just updated. We just forgot to add that direct evolution process.
It's okay if you can explain me.
Okay, I will explain it, sir. Like we have to extract the plasmid. That plasmid is there, no, PHY AMYL, by using by using alkaline lysis method, and then introduce genetic variation via mutagenesis using of error-prone PCR to introduce random mutations. And then after that random mutation, we just comes under clone modified.
So you said error-prone PCR.
Error-prone PCR.
Okay. For random mutation.
Random mutation in alpha amylase.
Okay.
By using Taq polymerase which are altered magnesium or manganese concentrations, and then the run around PCR run around 25 to 30 cycles for a mutated variant.
Okay.
And then clone modified genes back to PHY, that plasmid strain, I mean plasmid, by using of some enzymes, restriction enzymes to linearize the plasmid, and then coming back, random mutated gene is there, no, just by using the T4 DNA ligase, we will ligate to plasmid, and then that transform the updated plasmid into the E. coli for amplification, initial amplification, and then by using plate transformines, by using LB agar, it is like a lysogen agar broth, by using with antibiotics such as kanamycin, and then extract the amplified plasmid to the plasmid, and then transform that to the Bacillus licheniforms, and then prepare. Again the same process comes, like plating the transformants, and then with selective agars like LB agar with antibiotics, and after that screening method come. Screening is like with starch iodine assay. By using of that starch iodine assay, we will identify the significant starch hydrolysis, like color will change, disappearing of blue, black color. So after that, secondary screening process happen, like testing of enzyme activity, thermostability, and then pH tolerance and catalytic efficiency. First test thermostability, like incubate enzymes at 70 degrees Celsius for 30 minutes, because it is I am using that Bacillus licheniforms because it produces the alpha amylase in the culture media, so that I am using incubating enzyme at 70 degrees Celsius for 30 minutes. And then by using, measures the residuous activity using DNA assay method, and then comes under pH tolerance. pH tolerance, it is test activity in buffer ranging from 4 to 10 by using citrate, phosphate or tris HCl, and coming back to catalytic efficiency by using performance of Mitchell's-Menton kinetics, growth kinetics, by using that. Yesterday I think you informed that VM or KMs, VM, Vmax to that one, and then coming back to the fermentation process, we have used first we have used the initial batch, and then fed batch. This is a continuous stir tank reactor, followed by Nikhil.
So the fermentation process we used was the fed batch fermentation process, which was a stir tank fermenter operated in fed batch mode. So we took initial batch phase and then fed batch phase. The initial batch phase was about 12 to 24 hours, which was operated at 7 pH and 37 degrees Celsius temperature. And the next was a fed batch phase, which was operated for 24 to 72 hours. Next, the upstream processing and the fermentation part is done. Then we went to the downstream processing part. The downstream processing, first of all, includes filtration. As he told earlier, that our product is extracellular, that we don't want the cell, our product is in the broth itself. So first of all, we filter the cell and biomass that was all collected in the broth. So filtration, with the help of filters, we filtrated all the cells that are there. And then we went for filtration and then centrifugation. We want to, all the heavier particles, we want to remove them, with the help of centrifugation. And then we went for purity ion exchange chromatography. Now we would have gone for affinity chromatography, but that's pricey. So we went for ion exchange chromatography, and our product, which is alpha amylase, is at pH 7 is negatively charged. So we went for a stationary phase, which is positively charged resin. So through ion exchange chromatography, we went for purification. And for concentration, we went for concentration, ultrafiltration, and stabilization. Concentration, again with the help of concentration, then ultrafiltration to the help of ultrafiltration membranes, and stabilization with the help of compounds which are inexpensive like sucrose. Okay. And then we want the product to be in powder form, okay, which is more lasting. So we want to increase its shelf life. Okay, so we went for conversion to powder form through spray drying, like lyophilization. Okay, and with the help of quality check, and then we went for packaging. Next slide, please, sir. The next slide, the last slide, like explains the costing all of this. So first of all, it includes the capital cost. The total capital cost is 2.1 CR. Now it might look very big. It is because it includes everything. Like in the first session, you told about no one mentioned out the land and etc. So 2.1 CR includes the everything price. Now let's say we are supposed to pay it in 10 years, amortized. Okay. So our cost becomes 21 lakh per annum. Okay. Then the capital cost per kg becomes, we are like operating 50 kilos, 10 kilos over 500 batches over the year. Okay. So the cost becomes 420 rupees per kg. Okay. So operating cost. Then we go for operating cost. Our raw material cost is 14,25,000 for the entire year. Utilities 15 lakh, labor 37 lakh 50,000, maintenance 7 lakh 50,000. Then the total operating cost comes out to be 74 lakh 25,000 rupees. And the operating cost per kg comes out to be 1485 rupees per kg. Okay. So total cost per kilogram comes out to be 420 capital cost and the operating cost 1485. So our total cost comes out to be 1905 per kg. And also compared with the one available in the market, that was 1990 per kilogram. And that was also for starch processing grade. It was also not good grade like pharma grade. And that was we have prepared. Okay. So if we see the revenue production, so like do the analysis, then in 2.2 years only, we can pay back all the things like 2.1 CR we have assumed. That's it, sir.
Okay, and this 2.1 CR included the R&D
everything.
part also?
Everything, sir.
If I give you that much money, you will work on this?
Sir, obviously it's a theoretical model. If
let
you
me,
give that,
let me go
definitely I
back,
will
look
work
into
for
my
it.
bank accounts. If I have enough money, I will come back. We'll invest in
Sure,
this, okay?
Sure, sir.
I like the idea. Now, see, basically my intent with these two case studies for you was not only to work in a team, you get to work with multiple people with different backgrounds. But the point is that it's easier to know or learn about a new technology. So I can teach you, let's say I taught you synthetic biology. If I can start teaching you molecular biology, I have to prepare a little bit, but I can do that. I'll teach you all the techniques and everything. We have never done that in the lab. You have no hands-on experience. When somebody comes, they give you a speech, they will say like, okay, these are the things that we did, this is how we can approach establishing an industry or developing a product. Product development requires a lot of understanding, not just about the product, but about the process that you have to go through. But it also has to have who's going to be your end user. That's the key. When, let's say, if my industry is developing the straps for the watch, who would be my end user? Is it the public or is Apple? If I am developing the strap of the quality that Apple requires, my end user is Apple. Once Apple buys it from me, what they do is up to them. Their end user is you, the customer, right? So depending upon whom you are targeting, your process development changes. The amount of capital that you are going to invest will change. So having that entrepreneurship is good, but having an understanding of how to approach it is very, very important. Understand the value of your product, understand the market size of your product, only then try to implement it. Location we call, right, for when you are trying to establish a particular industry. If Rourkela steel plant was not established here, established somewhere else, let's say another 1,000 kilometers away, where they don't even have access to railway or road or the iron ore, they will not be a profitable company, right? It has to be that way. When you are establishing with respect to your feedstock, you have to make sure you have access to your feedstock. Theoretically, in an experimental world, as part of my research work, I can say, hey, I can use agricultural feedstock for production of this, but I have to have the capability of utilizing it on a large scale. If I don't have that capability, the technology is not worth for me. Sima? I hope you learned something from me. This is the last day I am here. I will leave pretty soon, but thank you so much. This would be it. Thank you. Yay.